Hello, everybody. Welcome to the Reptile Rejects. I'm your host, Michael, and this is Dominic, and we're the Rejects. Alrighty, Dominic. What has been going on for you the last week?
This week, one clutch has been hatching. My second clutch of pygmies has started coming out. Actually, there's still a half. I think I only have one or two out, and a couple of other ones still coming out, which is definitely something I want to talk to Dr. Julander about. See if he can help me finally maybe get some of them feeding before they all perish. Except for that, this week, not that much. I'm expecting our big import to come in pretty soon. I ordered a lot of GTPs and I am very impatiently waiting, and it kept getting delayed. It's been 4 months delayed, so supposed to come in tomorrow at 10 o'clock. We shall see, because it keeps getting bumped. So, except for that, that's probably my news this week. What about you, Michael?
Well, nothing too new, just mostly work stuff. But I think that Bahama Boa might be a little gravid now, as she's looking pretty thick. Hopefully, she didn't just like ovulate this week, and that's why she shed. But she was looking awfully uncomfortable right before she shed this week, and she still ate yesterday. But I don't know. She's looking oddly big for not eating as regularly as I was feeding her earlier this year. So yeah, hopefully, fingers crossed. Obviously, could just be a female full of slugs, but just fingers crossed that there's 30 little tiny boas in there. And yesterday I ended up pulling the trigger on a pair of Angolan pythons for my buddy that I think are produced by Don Patterson. So I was like, I've been debating those for the past like 2 years, and I was like, I might as well give them a try. Every time you look at them, they're just so you just can't stop looking at them. So decided to give them a try, and I'll pick those up down in Calgary in August at the WCRE. What about you, Justin? How's your last week been?
I'm pretty busy, but good. I like Dom. I also got another clutch of pygmy pythons hatching out, so they're still sitting in the incubator for another day or two before I can bring them out to the reptile room. I like to kind of let them sit out there for a bit, but um, first clutch is shed out, and I've started feeding trials, so that's always a joy. So yeah, but other than that, just had a water polo tournament yesterday, and Friday, so kind of a busy weekend. I'm a little bit tired. Hopefully, I'm not too off my game tonight, talking with you guys. But yeah, thanks for inviting me on. Oh, I can't hear you.
I mute myself when I'm not
Yeah.
when I'm not talking to try to avoid any extra noises. But yeah, that's it's always awesome to have guys like you on a podcast because you guys you're a wealth of information and you've written several books now. So yeah, it's always good to have you and Mutton and all those guys on.
Well, yeah, it's fun to do shows, and always great to have more podcasts, reptile related podcasts to listen to. So yeah, I applaud you guys' efforts to start this up.
Thank you. Hopefully, we can do a little bit of justice and try to be at least as half as good as you guys in NPR. But we'll see. Only time will tell.
We got to
Alrighty,
see if we can
Dominic. Do
beat
you?
the. We got to see if we can beat the seven podcasts. Isn't that the average? People do seven podcasts
Right.
and then stop.
Yep.
So
Yeah.
Yeah. Yeah. It's can be hard to keep going for sure. Like you know, I see why a lot go out of just kind of stop doing it after a while because life gets in the way and stuff. But yeah, you really got to put in the effort to keep it going strong, especially NPR guys. I don't know how they've done that so long. Very impressive.
Yeah, they've kind of deserved taking a bit of a break last year or so of
Yeah.
being not as regular, but it's understandable. There's
Yeah.
500 plus episodes to rewatch, which I've even gone back on. What was their old platform?
Oh yeah.
What was it?
Um,
Not I'm thinking StreamYard, but no. It's the other one where I've gone back and I've found like Rob episodes from 2011 or something when he's talking about something that I didn't even know he talked about, and it was like he was talking about diomes in like his
right.
first episode on NPR. That's pretty cool, but yeah.
Yeah, it's Eric just amazes me the amount of work he does. He's the one that kind of edits ours and puts it up and stuff. So we just have to record and he does the rest. So yeah, that guy. And with his schedule, his work schedule is really busy, and he's up at the crack of dawn and applies the hood to go to sleep, fairly early. So yeah, I don't know how he gets so much done, but he's the man.
Yeah,
Yeah,
definitely.
it's pretty cool that he does
Oh,
that for sure. Is
Yeah,
that my phone that keeps buzzing, or is that somebody else's?
it
Or
seems
my
like
deal?
it pops up when you talk. So I don't
Oh, is
know.
it mine?
Yeah.
yeah.
I
You
don't
can
know.
always just mute yourself when you're not talking, and
Uh,
it should be
I
fine.
don't think it's me. Though.
Or maybe it's you, Justin. Sometimes I do hear it on the Fight Club, but it's all right.
Oh, really? Yeah.
I don't know. I'll try to figure out how to get
Technical
rid of that
It
issues.
might be your email.
What would it be without? Yeah.
without any issue.
Yeah.
So, Justin, our goal today would be to get three subjects in. Probably talk a bit about the inlands, talk a bit about pygmy feeding, and definitely talk about that research paper that I was trying to find anybody to talk to about it because nobody really understood it or knew what the heck it was, and made the mistake of never reading who actually wrote it. And so I ended up asking Justin, I'm like, oh, could you come on and chat about this research paper? I bet you'd understand it. So Justin ends up opening the paper. He's like, yeah, I know this paper. He, being the co-author, I figured you're probably a good person to talk to. So I'd love to chat about that. We'll probably keep that maybe for the second half of the podcast. But
Sure.
before we get started, Michael, do you want to do just maybe I don't know, maybe a basic intro of who you are? Nothing too long because everybody's heard your story probably about a million times. But I don't know, give us a 3 minute of who you are, your qualifications, what you keep, anything you want to share with the group.
Sure. Yeah. Justin Julander. So I'm from Utah originally, and I've lived here most of my life, and will continue to live here as long as I can. It's a great place to live, beautiful state. Kind of just like most people that are listening, I had that reptile bug really early on. Kept as a kid as much as I could. I had very supportive parents, so it included a lot of cool reptiles and mostly native stuff, but also a few exotics growing up. And then after I got married, got really into Australian reptiles. I mean, I was always kind of interested in them, and then started Australian addiction reptiles and started my Australian reptile breeding career, and then just kind of as a hobby side business type thing. But I went and got a PhD in virology, so I study antiviral research, how to kill viruses basically, and um, that's been really really great field until about this year when the federal government decided to stop all funding for some unknown reason. So might have to come get a job up with you guys and help fight viruses in Canada instead of the U.S. if they don't figure things out here soon. Um, so my qualifications, like research wise, are mostly in the microbiology field, but uh, I've tried to study reptiles, and sometimes that gets in the way of my real job. I spend a lot of time thinking about reptiles, and it was always kind of the dream to be a herpetologist. So, I kind of fulfilled that to some extent in my brain by writing books and kind of making that bridge between the scientific publications, scientific community, and the keeper, the hobbyist for the most part. So that was kind of the goal. I don't know if I achieved that, but I've written a few books with some great co-authors, and um, uh, the Carpet Python group was kind of my first love in that regard, and that was the first book that I wrote with Nick Mutton, and then we put out a second edition not too long ago. But I've also written books on the children's python group, the Antaresia genus, and the green tree pythons. I wrote a kind of a smaller book that's on Amazon, and then a knobtail gecko book. And is that it? I think that's all. So working on a couple more. I've got one coming out soon that's a field guide to the reptiles and amphibians of Utah, and then working on an Aspidites book with Lucas Lee and Nick Mutton and Zach Loafman. So 4 co-authors and 2 species. So we'll see how that goes.
Well, that should be really cool, actually,
Awesome.
especially the Aspidistis book. I look forward to that one.
Yeah, they're fun.
How about your
Even that
collection wise? Oh, go ahead, Michael.
even that Utah book should be pretty cool. Honestly, Utah has some. It's a weird state, right? Because down in St. George's, you got that just a little bit of Arizona and Southern California species with the lyre snakes and the cal kings and
Yeah,
all that fun stuff. So
yeah,
that'd be a super neat book to read.
yeah. It's got a diversity of landscapes. Really great deserts, mountains, all sorts of good stuff. So yeah, there's a lot of cool reptiles here. It's kind of a double-edged sword because it's nice to have it. Not everybody know it's like a really great herping spot or herping location, but I guess we got to share. Everybody thinks of like Arizona, that's the place, and it is a great place to go for sure. But Utah's got some pretty phenomenal landscapes as well as some amazing reptiles. So
For sure. Yeah, it's like here where not too many people know that there's such a diversity, especially in the interior. So when when you go out herping, you're like the only people out there, unless there's some car meet thing happening in like an area with no cell reception or however it is. Or but it's nice going up and down dirt roads and there's nobody there. So yeah,
yeah,
makes it easier to herp and you never have to see anybody.
for sure. Yeah, Arizona's great, but you also got the risk of running into drug traffickers or illegals or whatever, and that could be a sketchy situation too. Or border patrol, that could be sketchy as well. Trying to
Definitely.
convince them that you're not a coyote or something, you're just out looking for snakes.
I could see
Yeah.
that.
Alrighty, Dom. Do you want to start off with the first question?
Sure, but well, just before, obviously, we want to talk about inlands a bit. Do you keep any inlands? Do you have any? Have you bred them?
Yeah, yeah. I've had inlands for quite a bit and really enjoy the species. They're just phenomenal snakes. One of the calmest, or probably the calmest carpet python by far. That's the species I can usually just kind of reach in the cage and grab them, and they're not going to tag you or light you up at all. Um, but they're really beautiful. They almost people would probably think they were axanthics or something. They're kind of black and gray, but they have a really cool blue undertones that are hard to pick up in photographs. Yeah, when I first kind of saw the pictures of them, I thought, they look like kind of a, maybe different looking coastal or something. But yeah, very different than a coastal and very unique in their natural history and just a really cool kind of desert specialist living out in the harsh areas of South Australia and into Queensland and New South Wales. Really cool species.
Very nice. Do you want to talk a bit about maybe the different lines that we have in available in North America, and also the what's the different type of the one that's pictured on your front page of your book? Explain what that type of inland is a bit.
Sure. Yeah. So I guess that's the. So yeah, the lines we have in North America include a few that have been brought in from Europe. The Schofield line. Frank Schofield was a amazing breeder in Europe. Produced a ton of different python species, and then the Maug line. I believe that one pop. I'm not the best on like the history of the lines and stuff. That's more of a Nick Mutton question, but so I've worked with both of those lines, and then I imported a pair from England, and the male was unrelated to any other line. So the female was a Schofield and the male was unrelated. So that's kind of what I refer to as the AAR line, which is kind of a Schofield outcross with an unrelated male. So they're kind of half related from any Schofield line and fully unrelated from the Maug line.
Just by curiosity, do you know if that derived from UK Python through him? Because I have one that is 25% Schofield and 75% an unrelated line to the Maug or the Schofield, And basically, he didn't really want to say how it got there or which way, but he just said, "Trust me, it's not Mog,
Mhm.
it's not Schofield, it's another line, and it's completely unrelated." So I
Yeah.
don't know if that would be it obviously it's from Europe, so somewhat the same destination, but or same area.
Yeah.
But
I mean, I think, in herpetology or in herpetoculture, you'll kind of find that where, especially with things that are, a little, not supposed to be exported from their native land, you'll see things come in through like Czech Republic or Germany or things like that, and so you'll have kind of one-offs where you'll mix them in maybe to known lines because that's your only option when you have kind of one that's unrelated. And I think that was the case with this one. I can't remember the exact story, and I didn't find out until I actually got them in the U.S. Like he said, "Oh, that one's unrelated to anything else." I'm like, "Oh, okay." So you can kind of see what the implications of that are. But I probably wouldn't have brought them in if I would have known that in the first place. But kind of a happy coincidence after the fact. So I just thought they were Schofield, kind of like that line that Paul was working with. But I guess kind of like, okay, that works. I can work with an unrelated male. So, and he's still around. Not really productive. He's a little older, but yeah, his I've got kind of like second generation offspring from him, and that Schofield female. And then this year was the first year I bred, and one of these offspring from the unrelated male and Schofield female to a Malgline female or male, Malgline male to an AAR line female. So I don't know that there's too much difference between the two. I don't know that you can look at one and go, oh, that's definitely this line or whatever, because I think there is a lot of overlap in kind of the color and pattern. I think a lot of people think maybe the Malgline has more reddish undertone, coloration in the pattern, but I don't know. I don't see it. I don't, and granted, I had a pair of Maugs and a pair of my other ones, so it's not like I have a huge diversity of both lines. And right now I've got probably 6 pairs of plus maybe a couple holdbacks that just to have the project. So yeah, that's kind of the what's going on with inlands in captivity. But also, yeah, the cover animal. I saw that picture and I just couldn't get those things out of my head. They're from kind of the inland. So South Australia up through to maybe into Queensland, like Dejara and Longreach, that kind of that north or western Queensland area. It seems like they kind of run in a narrow band from South Australia all the way up north. It's hard to see. We're basing. Nick and I kind of did like a not necessarily a revision, but like looking at the genetic work that's been done by others. Also, I kind of had this epiphany of like carpet pythons travel with trees along creek banks, and I thought, well, maybe that the diversity or the genetic diversity of carpet pythons has something to do with the water basins in Australia. And I kind of overlapped what had been done genetically with the water drainage basins of Australia, and it overlapped pretty well, pretty closely. So it makes sense that they'd kind of be separated by different drainage basins. It might be hard to exit one and enter another, crossover. Not impossible, but maybe more difficult. So that was kind of the thought in that regard. So excuse me. These in the we kind of refer to them as the gammon ranges or flinders ranges, carpet pythons, and the flinders and the gammons are mountain ranges that kind of go from South Australia, north of Adelaide, up to fairly far up until it starts getting really dry, deserty habitat in kind of way inland. The Strzelecki Desert up in northern South Australia. They're fairly difficult places to make a living in, the gammons and flinders. I did a trip there to look for these carpet pythons. I went out with Jordan Parrott, and we spent a couple weeks out there. Also, a couple others joined us, Steve Crawford, and oh man, I'm terrible with names. I'm sorry. Uh, It'll come to me at some point. But anyway, we went out looking for the these carpet pythons, and did a few different canyons within the Flinders ranges, and then headed up to the Gammon ranges, and spent some long hard nights herping, and with no luck. We didn't find any carpet pythons. We saw about 40 shingleback skinks, so that was cool, and a few different dragon. I saw a few lifers, Gidgee skinks added to that to my life list, and we saw Thorny Devil in South Australia. That one was pretty cool, but so it was a great trip, but just no carpet pythons. And then incidentally, Jordan went back with his family the following, so the same year maybe, or the following year,
And
and
found some.
found one, and it turned out to be the same one that was on the cover of the carpet book. So just a great story. Yeah, he texted me. He said, "Hey, check out what I found." And he said, "Can you send me a picture of the cover of your book?" Because he was, had very little connection out there. But so I sent that over, and he's like, "You'll never believe this, but it matches completely." So it was pretty exciting.
Just by curiosity, did you guys ever do any genetic testing to compare the inlands either in captivity or inlands in Australia compared to the gammon range of the Flinders? Was there any genetic diversity or even like, I don't know, like a percentage of difference, or were they basically the same thing?
So the genetic work that has been done on the carpets was a paper that was basically looking to see if they could. I mean, it started kind of from a paper looking at, or it was actually a dissertation. I don't think it was ever published, but it looked at the diversity of genetics within the carpet clade, to see if there were ways to determine if something was, caught from the wild, or mainly to see if they could see where the carpet originated from for punishing wildlife crimes, or to see if something was illegally exported or something like that. So that was kind of the idea behind the genetic work, for kind of law enforcement purposes or wildlife enforcement. So they were looking at kind of how to differentiate the different carpet pythons. So they had kind of a wide array of genetic work and a lot of different samples. And then that work was continued on with a published paper. And of course, the names are failing me. I could probably get it on my uh, folder that I used to write the book, but it's escaping me right now. But they had done even more specimens of carpet pythons from around Australia, and kind of laid out the how everything was arranged. And it looked like there was, some of these specimens from the gammons flinders grouped a little differently than the typical inlands from New South Wales, and the other specimens that they had there, and that lined up pretty well with a drainage basin type overlay. And it just looked like there were potentially some differences genetically there. Phenotypically, they're somewhat different than a typical inland carpet, and so we kind of hypothesized that they could be separate subspecies. Maybe inland carpet pythons are a species, and with a couple of different subspecies. So we would probably suggest that might be the case. But again, we're not taxonomists. We did some work with Warren Booth, who is a taxonomist, and knows that stuff better than we do. And he did some analyses with some samples that we collected from captive animals with known, origin or locality data on them, and looked at the different how things shook out. And it was comparable with this paper. And so, you know, diamond pythons are really different genetically, and they also seem to kind of align closer with Centralian carpets, Morelli Bradley. So it's kind of an interesting thing, but what do you do with that? I don't know. It's hard to say. Is Spilota different, subs or different species from, say, a coastal carpet or a jungle carpet or something like that? So we made kind of a different taxonomic proposal that kind of kept some of the previous subspecies designations, but added in kind of the gammons flinders ranges as a separate entity to whatever extent they are. Then the inlands, the southern coastals, and then we grouped the northern coastals together with jungles below the Black Mountain Corridor, and then the carpets above the Black Mountain Corridor up through New Guinea and Darwin are kind of in this, kind of that variegated clade, and they could potentially also have a couple subspecies, maybe. But I don't know. Taxonomy's goofy. That was just kind of what we came up with and stick to some extent, but not to say that that's the end all be all for sure. And definitely more research is needed to kind of piece things together. I did hear that the group that rearranged the Antaresia group is also working on carpet pythons, so it could be a lot messier in the future.
They're going to put everything back together, just like the children's.
Right.
So let me ask the question differently. So I know, according to the new book, Darwin's and, let's say, West or Papuan pythons are somewhat the same species now, but I'm pretty sure you would never breed your Papuan python to your Darwin, right?
Right.
Okay, that would be blasphemy. Now, would you breed your inland to a gammon range?
Oh, definitely not.
Okay. Yeah. So then would you put them at more the subspecies status then?
I think there's enough difference there. At least, I don't know. I like locality stuff because I want to see something in captivity that reminds me of what I saw in the wild, to the extent that I've seen them in the wild. This past trip, we got to see a Bredl's in the wild, see a Centralian carpet. So that was very exciting, very cool. And, I don't know, that's maybe a bad example because there's only, they're a different species and they're pretty isolated and they're not overlapping with any other species. But like, say, diamonds and coastals are they interbreed in the wild. Frankly, I mean, anybody can do whatever they want with their pythons. Once you get them into a box, it's not like they're going back to repopulate the wild. They're a pet. They're in a box. If you want to make a pretty snake, breed whatever you want to whatever you want. I just kind of like the idea of localities. That's what I prefer. But I've also seen some extremely beautiful snakes, Don Patterson's included, that are 3-4 way crosses. They're just amazing to look at, and I wouldn't mind having those either. So I'm not so strict that I'm like, I will never keep anything that's not bred to only locality or only its own subspecies. But that's kind of where I've picked my battles. And so
I struggle with that.
My buddy just got one of those big, he got one of those hold back adult non-jag, but it's a bredli diamond jungle thing that's like 7 feet. That looks, it looks like just a normal, like the classic, like bright yellow diamond jungle, or no, it is a jag, I think, but it looks like that classic diamond jungle look. But right. Then he's like, oh no, this is bredli in it too. It's like, I wouldn't have even known. Yeah. Right. So yeah, that stuff's neat.
Oh, they're incredible.
In the last book, you had Imbricata. Actually, you put the gammons in Imbricata. I only have the first edition, so I was just like looking at it. Obviously, it's been updated, and you did put gammons in the new one. But the other one, you had them in with Imbricata, and obviously that's a lot more of a split than what you currently have them in. But you guys based that off of information at that time, correct?
Right. Yeah. And that's, all you can do if that's all you have. And there is some resemblance, and who knows, maybe Imbricata and an inland kind of met up and they split off or something. Who knows? Like, it all this is old stuff that's happened long time ago, and we're just kind of trying to put together a snapshot in time. What is it right now? Because there is some evidence that there used to be joining uh, water systems from this area, the Gammons and the Flinders, with the rest of New South Wales. Those river systems, the Murray Darling River system that used to connect in with, I believe that's the story, that it used to connect in with the Gammons and Flinders, but then some geological event changed that, and it kind of isolated it. So it's potentially that there were inlands all through there, and then this geologic event happened, and then it separated them. And who knows how long it takes to make a new species or to have enough change occur to call a new species. Obviously, the current genetics just have inlands as kind of subspecies, but most taxonomists don't really believe in subspecies, so they just call them all Spilota. So they don't really differentiate between a diamond and an inland and a jungle, just as regional variants of the same snake, which is fair. Whatever. I mean, that's why I have the frustrations with taxonomy. Is it's like kind of like it's based on certain things, but there's no rules that apply to everything. So whoever's doing the research kind of makes the call, similar to that Antaresia paper. I disagree. I don't think Western Stimson's pythons are children's pythons. There may be some genetic similarities, or there may be some overlap in genetics, or there may be some overlap in range. That doesn't make them the same thing, and so that's been kind of a frustration, is like, you can publish things and there can be support for that, but that doesn't mean they have to be accepted. Easily the paper could have gone the other way and split them out to additional Stimson's types or children's types, because there does look to be a biogeographical barrier, kind of along the border between Northern Territory and Western Australia. And there does, those genetic differences on either side of that biogeographical barrier, look pretty substantial. So children's pythons in the Kimberley don't necessarily match up with children's pythons in the Darwin area, and so, or Kakadu, those children's python populations. Whereas some of the eastern Stimson's populations are probably align better with those Kakadu children's, or whatever, that stretch down, and they just kind of change phenotypically as they run south. So the authors took that to say, well, then they're all probably the same, because there's no way to really piece this out, and they were trying to make it fit with what was maybe published before. I don't know, but to me, their genetic analysis made it look like there could be additional, like, children's population, like children's subspecies, similar to what they did with the spotted pythons. But instead of saying, oh, these genetic differences, they said, well, they overlap in range, so they must be the same thing. But I'm thinking, I don't know if I agree with that. And, the Western stems overlap with pygmy pythons, and, there's definitely some broader genetic differences between pygmy pythons and Stimson's pythons. But, they just kind of went one way when they could have easily gone the other way, it seemed, from my limited understanding, but also conversations with real taxonomists who went, yeah, this is a head scratcher. I don't understand this either.
Yeah, it'd be very interesting to see how, those escarpments children's compared to even the ones near them, like whether they are just the same and somehow physically look like baby Oempelli pythons or not, right?
Yeah.
It's just super neat how right populations within, I haven't been there, I don't know how close they are, but you have. So within the certain, let's say 10 kilometers, how they're just so different, and you would think it's totally something else. So I'm sure that could, well, I just assuming that could probably be its own subspecies or even species, maybe. Yeah.
Yeah, I mean, it is a little difficult to piece those kind of things together, because, habitat definitely influences pattern and color and things like that. And you have some species that are wide ranging that have different phenotypes. And so that's kind of why I like the idea of localities, because if you keep a project to a locality, you're not going to end up crossing it with other things. I mean, a great example is green tree pythons. Everybody knew northern and southern green tree pythons were different. There were obvious differences in their phenotype, their structure, all sorts of things. And then, but people just kind of said, oh, they're all Morelia viridis, and we're just going to breed them all to each other. And you had all these Arubiacs and crazy crosses and stuff. And then it turns out, okay, yeah, they are different species, and there's enough genetic, and then now there's several subspecies, and so you've got all these nifty looking
Hybrids.
intergrades and hybrids and all sorts of things. But I just, my own preference is I would rather see a pure Aru versus an Arubiac or whatever. Not that Arubiacs are ugly or not desirable or anything. It's just my preference. So I don't know, but yeah.
I was going to say 100% agree. And you know what my frustration is? If you breed a coastal to, let's say, I don't know, a Bredli, it's non-desirable and it's worth nothing. You won't be. It's not going to be. It's going to be hard to sell. If you breed a Papuan to a coastal, again, same thing. But for some reason, in the green tree python world, you breed a Biak to, I don't know, a Manawarki or a Naru or anything else. All of a sudden, they're designers and they're worth 3 times the price, maybe even 4. It's one of those things I've never understood. Why do hybrid green trees fetch such a high price for being hybrids or being mutts or being like extreme mutts, but it doesn't work like that in our world? Maybe it's a little different for, I guess, maybe the really crazy jungle jag mix and all that. For example, what Don Patterson is doing, or there's a couple of guys in the States that do the diamond crosses and all that. But overall, I find that in the carpet world, as soon as you hybridize or you mix it, it actually brings down the value. It doesn't seem to bring it up.
Mm-hmm.
So
yeah, I maybe chalk it up to marketing. I don't know, because
fair enough.
The green tree python community kind of collectively, or the majority of them are kind of into the designers, and so they've marketed it as such. Oh, these are designers. We are trying to isolate these different phenotypes and make really extreme examples. And granted, there are some really extreme examples and some really interesting looking snakes, and you'll see that especially with hybridization. Some of the most interesting snakes out there are some that are hybrids, like a Borneo bat eater, kind of that crossing a Burmese python with a reticulated python. Very interesting, neat looking snake. Might be a neat pet to have or something, but it's kind of a one-off. I don't know why it would be more valuable than either of the locality or parent species, but it has an interesting look. It's different, and I think herpetoculturalists often go for things that are different and exciting or unique, and they want to say, hey, I've got a Borneo bat eater, and everybody's like, ooh, what's that? And that kind of thing. So and I think you don't know what something's going to look like until you try. And the fact that you can breed a Woma python to a ball python, which are arguably the more derived and the most basal species that are of python that you can find, and that they can make an offspring, is just incredible to me. It's like breeding a human with a pygmy marmoset or something and getting some weird hybrid out of it. It's kind of crazy. But there's all sorts of interesting things you can do in herpetoculture, and it's just not necessarily for everybody, but I do think there's some really interesting, neat looking hybrids. And you also have usually like the second generation hybrids are the ones that are kind of mind-blowing and really crazy looking a lot of times. That I don't know, I kind of agree, like for the most part you don't really get anything that's more desirable than either of the parent species, but sometimes you get some really crazy looking things that are like, wow, that's eye-catching, or that's very interesting, and some people might prefer that. I don't know, it's to each their own, I guess. But for me, I'll stick with the pure species or subspecies to kind of. I guess there's kind of the morph craze kind of blurred those lines a lot.
Yeah, it really did.
Everything was fine in pursuit of that morph, so you could breed whatever to whatever to produce that morph. And the consequences be damned. But that does have some bearing on what we might have available in the future. But I do think for the most part, there are people out there that care and will, continue to breed the species or subspecies or whatever localities pure and keep them what they might look like. But even in that aspect, like nobody's keeping the ugliest to breed with the ugliest. They're always selecting the most nice. And so you're going to kind of and introduce some morph uh, idea in there because you're selecting for a certain phenotype, even though there's a range of phenotype. I mean, jungle carpets are a great example. You see a huge variety of jungle appearances of jungle carpet pythons, traditional jungles, I guess. And they're you saw that with some of the localities that came into herpetoculture. We had Palmerstons and Tullies and all sorts of stuff that popped up from Europe, and people would get them and go, "Well, that's neat to have a Palmerston, but they're kind of, they've got a cool stripe, but they're kind of ugly. So now I want to get some color into that. I need to get a nice yellow Palmerston, and even to complicate it more, like I found out there was one spot that was a great herping spot because you could find jungle carpets pretty reliably there. But then it turns out any wildlife officials that were confiscating any kind of jungle carpet, they would release it there. And so then, like, okay, where did this come from? Is it a Mission Beach, or is it an Eddy Bay or whatever? Like all sorts of different areas in that tropical region where they could have come from, and they're all just dumping them on this road outside of Cairns or something. So it's like, you know, hard to
Just by
say.
curiosity, how did Jolitons end up being called Jolitons jungles? They were called. I still see people calling them like that all the time when they're not. Right? They're variegata. Is that right?
Well, yeah, I mean, I guess again, phenotypes, they do look like some of the other, this goes to kind of the natural area that they occur in, shaping the pattern and color and things like that. I always maintain that if you took a clutch of coastal carpets and dumped them in the darkest jungles of Queensland, you'd probably end up having something that looked like a jungle carpet, that black and yellow kind of pattern down the road. But so anyway, I think they just have a similar habitat, so that shaped them to look similar to those things. But that line of the Black Mountain corridor, that biogeographical barrier, it definitely seems to show genetic differences below and above that. And that's not just for carpet pythons, that's for several different species. It's kind of that line between the typical stuff from New Guinea that ranges all the way up in the Cape York Peninsula, and the stuff further south, in Cairns and that area there in of northern Queensland. Again, these are kind of maybe these biogeographical barriers aren't as important anymore now that we have roads and plantations and things that maybe cross that, or but usually those biogeographical barriers are because there's like a rocky area that's really hard to get across, for a tropical python that's not going to crawl across a bunch of lava rock or something in the middle of the day, or it's not appropriate habitat for them, or the tree lined creeks don't run, that north to south, whatever. So just kind of the way I picture it, I guess, in my mind is there's this area that's just difficult for genetic flow to get across, at least historically, but that may have changed with human intervention and human development and things like that. So that could shape and change things. Like I said, maybe some wildlife biologist is dumping things across that barrier, and so, but the idea is that those things up above that barrier align more taxonomically with the variegata complex, if you will. Again, that could be 3 different subspecies in New Guinea and then the Cape York Peninsula, and then over in Darwin. They're not identical. They're not the same, but they group together as kind of a cohesive unit to some extent. And that paleogeography, there was a land bridge between New Guinea and Queensland and also kind of Darwin and New Guinea. So there could have been gene flow as recently as 8 to 10,000 years ago. So that's kind of the idea why they might be as closely related as genetically as they are. So that's kind of the thought process we put into this, trying to group things and put labels on things, which is what people like to do. They like their labels. But I guess I find that the localities are easier to keep track of than the genetic differences and maybe what constitute a subspecies or a species or whatever. If you keep a locality, you're probably going to be safe. And I always go back to that, So the Julatten jungles are above that corridor, so they align more with the variegata, even though they look more like the stuff south of the border that's in similar habitat. But I think that's just the influence of habitat. If you go further up on the Cape York Peninsula, there's some over there that are virtually indistinguishable from a Darwin carpet or even a Papuan. So, it's definitely a difficult thing to put labels on, right? And that was kind of the idea that prompted that, is looking at, now that we have more access to photographs and to locality data for different animals in general, with the advent of iNaturalist and even like photo hosting sites. I find a lot of use looking through like Flickr, for carpet pythons. Then you can see maybe where they came from and get a sense for, okay, what do they kind of look like in this area? And I think back in the day we had a snapshot. Okay, here's a jungle from, Queensland and here's a coastal carpet, and this is how they're supposed to look. And then you go there and realize there's a huge diversity of appearances and patterns and colors and throughout their range. And to say this is what it has to look like or it's not a pure coastal just became ludicrous. Like you can't ID something based on a phenotype necessarily. We saw a jungle in southern Queensland, down in Brisbane area, which is, kind of those southern coastal carpet pythons. And it looked very similar to a jungle carpet. It had bright yellow bands, had a really distinct head pattern, things like that. So I'm like, if I was in Cairns area, I would say, oh yeah, this is a jungle carpet for sure. But, it was a southern coastal. So I guess that's also some of the evidence that they use to say, they're all the same, like carpets are carpets, So again, if you're breeding a locality from southern Queensland, you're going to be safe to say, I'm not crossing it with any other subspecies or species or whatever you want to call them.
Yep, fair enough. Do you have any questions, Michael, or do you want me to continue?
Well, I definitely think that's a very good, well spoken out explanation for the way you did your book and explained it. Definitely, that's like how gray band guys, right? They just locality. As long as it's that locality, whether they go somehow for some reason go back to Blair's, even though that's obviously probably not a thing. You got your 277, you got your 9 Mile North. It is what it is, as long as you stick with that. So yeah, that's super neat. I think Dominic probably wants to ask you about maybe sexing those male inlands. I think he might have some questions about that.
So I've got actually two venues that I want to go down. Number one, I want you to tell me how you breed your inlands. Do you do spring or winter, or kind of in between? I'm just curious about that. And after that, I want to talk about sexing inlands. So to see if you had any variations on inlands compared to all your other carpet pythons when it comes to sexing, either probing or popping. So if you want to go down those two avenues, that'd be great. Purely for, well, because I've run into issues and I'm really curious.
Okay. Yeah. I mean, I can give my 2 cents. And again, like with all this taxonomy discussion and that kind of thing, I could be completely wrong. I fully admit that, like I'm just going with what I have and what makes sense to me. And, I put as much work as I can into it and used as much the information that's out there. But so that's kind of my, I guess, disclaimer to everything I say. I guess that's the scientist in me. I don't like to make like distinct, this is how it is, like, because it could be all complete nonsense that I'm talking as well. So I fully admit that. But so I do things a little differently than most people. I don't just, introduce like every week or something in certain months. I'll usually just throw my stuff together in the fall, late fall, and leave them together pretty much all winter long. I'll take out the males when the females obviously grab it, and that's about all I do. So I keep it pretty simple, and try to. So I guess kind of back calculating, like my inlands have already hatched out, so they must have bred sometime in the late winter or sometime in the winter. They definitely weren't breeding in the spring and producing in the summer. So I do think you can breed inlands in the summer, or sorry, winter or spring. In the wild, they have a huge range potentially. They can occur quite far up in the well, not too far in the north, I guess, if we're considering those inland ones, the gammons and flinders types. But so yeah, I mean, it's pretty extreme habitat or extreme weather conditions where they occur. So I think that restricts them, just naturally to breeding in the spring, potentially, depending on where they're found and what conditions they have available. But you can definitely breed them in the winter in captivity. Lots of people have shown that and done that. So I don't think it's as restricted, or you're not going to have success if you try to breed them in the winter versus spring. But that also depends on how you're keeping them. If you're allowing them to heat up during the day and things like that, and they're just having a nighttime drop like other carpets, then they'll probably breed like other carpets in the winter. But
How low does your nighttime drop go?
my room gets into the 50s during the winter. In depends also on the cages. Like the cages lower down are typically a little cooler, and so I it, it, I've often housed my inlands in kind of the lower cages versus the upper cages. But this year I housed them in some of the upper cages, and didn't make too much of a difference. It seemed like they produced around the same time. And
I was gonna say, if you're going down to the 50s, though, that's more like Bradley Diamond territory. Though you're considering you're going much lower. Like you
yeah,
would.
and that's
Go ahead.
also like the extreme. Like it's not a 50 degrees for 10 hours. It's 50 degrees maybe for an hour or two, and then it's back up in the 60s, 70s, 80s. So it's cyclical for sure. Like it gets probably into the 70s during the day and down to the 50s at the very coldest part of night. So there's kind of I guess that's what you see in nature too. Like I was, this weekend I was sleeping in a tent, and it was 98 during the day, and at night it got down into the 50s. So it was 51 degrees, in the at the coolest time at night. And that's the same thing that's probably occurring in their range. In the daytime it can be really hot, and then at night it cools way off, and that's kind of the desert ecosystem. And that's probably why desert species like inland bearded dragons or leopard geckos or things like that do so well in captivity, because they can withstand a lot of different and extreme temperature regimens, whereas things they're tropical and are used to that steady 70 degrees year round might not do well in an extreme variable environment. But so that's why I think they're probably successful in a lot of different thermal regimes that you can use in captivity, and why they're pretty straightforward as far as breeding. As long as you're cooling them to some extent in the winter, you're probably going to be okay.
Yeah, I this year was my first time successfully breeding them, and I always bring my temps all the way back up during the day. I think I'm 77 or 75 ambient during the day, but the nighttime
Right.
I go as low as I can, and the lowest my room will go down is 64. So, but
Okay.
it was enough cooling down to make them go. So I was just
Right.
curious to see if you went lower, and if you go down to 50, that's I would consider that more of a spring breeder temperature than
Sure.
winter breeder for sure.
Right.
Yeah.
And
So
they were they'd still eat during the winter cooling, and because their lights came on in the day and they had enough. I mean, they reduced their intake for sure, and I didn't feed as often, but like they were still wanting to take a meal here and there, and so I'd offer it to them once in a while, even during the cooling period. So they were getting warm enough to digest food and that kind of thing. So yeah, it's again, they're pretty tough and can handle a lot of different things. And I think in the wild, it's just such an extreme environment, that they'll take every opportunity they can. And I think, in general, wild reptiles are probably trying to maintain a body temperature around the mid 70s to low 80s, so if it's really hot out, they become nocturnal, or they, seek shelter that's cooler during the daytime. And then if it's too cold, then they're more diurnal and they'll bask more and things like that. That's why it's a lot easier to find say a scrub python in the daytime in the winter, because they're out basking and they're congregating for breeding and things like that. Whereas in the summer, you got to go out at night looking for them and find them, cruising around in the dark. So just depends on the time of year, but they're kind of modulating their activity and their natural behavior to kind of keep that in that zone, I guess, so they don't overheat or freeze.
Oh, fair enough. So going back to the sexing, like probing inland. So have you noticed any either morphological differences or any changes for inlands compared to the other carpet pythons?
So I pop my inlands, and man, you can do that in the dark blindfolded with two left hands, and they pop pretty easily. Like the males will pop really readily, and they're very easy to sex compared to maybe like a jungle or a coastal, I found. And similar to Brettles too. Brettles are really easy to pop, and very obvious if you got a male or a female. You can almost tell from their tail shape to some extent when they're if they're a male, or I can usually kind of look and predict what it's going to be before I actually pop them. But yeah, I mean, I haven't had much issue with that. Although I did have one recently that I thought was a female, and I put in with a pair, and the two males started kind of doing a little combat. So I'm like, oh, maybe this isn't a female, and I popped it, and it was obviously a male. So, and I thought I thought I was female heavy, so it was kind of nice to have a male or a female turn into a male at that point, although that's not always desirable. So I was a little surprised that I got that wrong, and I'm not sure exactly how that happened, but sometimes I'm not perfect by any stretch, so that happens sometimes. But typically they're really easy to pop, especially soon out of the egg. Like if you wait longer and they have a little more muscular control, they're a little older, they've started feeding and things, they usually are more difficult to pop. So maybe that year I waited, to sex them a little later. But this year with the inlands, like I think I had like 16, 17, somewhere around there. And so I'll split them into a male and a female bin, and I'll pop them all like after I take them out of the incubation chamber, just to separate them into males and females. And that's what I've done over the last couple of years. And so, they've kind of stayed what I thought they were. Yeah.
And how about probing? So popping, I agree. Like I popped mine and they're very simple. They're no different. But probing wise, I found that females probe really, really deep. And I don't know if that's just me. I was just wondering if you've had that experience or anything like that. Like I've had females, like the one female that laid, she probes on both sides about 14, 16 scales down, which would everything would tell me that's a male. And she laid eggs, so I can tell you she's a female.
Yeah. Right.
So I'm just wondering if you've noticed any of that, or that's because I've been told that by like 2 or 3 people now. So I'm just wondering if there's any reason, because I've heard a lot of species differentiation is often based on the hemipenes. I don't know if that's so I'm just wondering if there was anything there for no.
I'll be honest. I haven't done a lot of probing because I pop them out of the egg, and so yeah, I don't really double check with probing for the most part, just because I feel like I can trust that if it popped, it's definitely a male, but obviously, but the females are pretty easy to tell as well. I guess it's really hard to missex them if you're popping them. So I haven't really double checked with probing. Um, so yeah, it's a good question. I think there may be something to that. Um, I guess generally, there's the females probe less deep, but it is possible to kind of pierce through the membrane, but that takes a little pressure to do so. So I don't think you're doing that. So there, obviously we should probably look into that and see if that's a consistent thing in different populations if their females probe deeper. Because I've also got a couple confirmed females that I could check with that have laid eggs, and I can probe them and see how deep they probe or whatever. But yeah, that's interesting observation for sure.
Yeah, I was just curious, and I've probed hundreds and hundreds of carpets, so trust me, I'm not going through them.
Yeah.
Um, cool. Well, I think we're good. Do you want to move to the pygmies? See if we change a bit of subject. So, pygmy wise, I know you've bred a lot of pygmies. You've talked about them. You seem to be one of the. There's not that many people who breed them, right? Like it's one of those things where people don't really like getting into them because there's such horrible, like there's just so much work. Sorry, let me rephrase that. I love the species. I love the
Yeah.
animals. They're one of my favorites. Okay,
Right.
but they are horrible to get started. So
Yeah.
I'd love for you if you want to go on your long rant and just talk about everything you do to from the hatching out of the egg to getting them to the point where you feel comfortable selling them to people. So I'd love to
Right.
know your technique, how you do it, how you get them feed, and all your little tricks. Go as much into depth because this is like my biggest struggle. I've bred them now. I've got two more clutches this year. I think this is my fourth year in a row, and I can tell you my success rate of hatching is 100% or 99, but
Mhm.
making them survive past 6 months, I'm probably around 10%. So I have
Yeah.
horrible success baking them. So I'd love to hear everything you do and
Right.
how you do it. So the mic is yours.
Sure. I mean, I think well, I had a recent interaction with somebody online. They messaged me and they said, "Hey, how can I get my pygmy pythons started?" And I said, "Oh yeah, well, I wrote a book on that topic, and I don't feel like necessarily typing it out for you, and you can read my book. That's why I wrote it, and the idea was, well, I don't read, so and I'm like, well, I don't spoon feed, so but I'm happy to talk about it on a podcast, and I have done that many times. I think they are tricky to get started because we are trying to start them on pygmy mice, lab pinky mice from a lab bred strain, and I think that's kind of what we're trying to force them into doing something they're not naturally designed to do. They're looking for lizards and lizard prey. And I know I've heard from Casey Lazick that when he bred some of the first ones in the country, he was trying to get him onto pinky mice, and he was just kind of pulling his hair. Well, he said the first year he did it, they all took to pinkies really easily. And he's like, these are a piece of cake. What a dream, and then the next year came around and he had to eat his words because they were all a nightmare, and he couldn't get any of them to take a pinky, and he was just really frustrated. And one day he kind of threw in a like a stillborn gecko or something like that. And before the gecko hit the floor, the snake had it wrapped and had eaten that thing. And so he started feeding them some geckos. I mean, they were obviously worth a lot more money back then. And so he wanted to keep them alive. And so he was willing to do whatever he could. And he said he kind of noticed like, and I believe this is true of the wild populations as well. Once they reach a certain size, they kind of have a dietary shift, similar to maybe a green tree python, where juveniles are eating mostly lizards. They're mostly diurnal on the edge of the canopy or whatever. And then as they age, they start to turn green, move into the canopy, and start feeding on rodents. And that ontogenic color shift corresponds with a shift in diet as well, moving from lizards to mammals. And I think pygmy pythons, because they're so small at hatching, all that's available pretty much are lizards. That's about this only meal that they can take down. And they're not going to go, like a baby pygmy python's not going to go try to battle a mother, rodent or marsupial or whatever it is out there, some kind of antechinus or something, to get a pinky antechinus. They're gonna look for an easy prey, which would be a small skink or gecko. And so, in Western Australia, there is no shortage of skinks and geckos. Like, they are everywhere. Western Australia is like the mecca for lizards and geckos, and of all sorts and sizes and kinds. So they have a pretty wide variety of things that they can eat. And also, they're also known as anthill pythons because they're found in termite mounds sometimes. They can take up residence in there. It's a nice thermally stable environment. Coincidentally, geckos and skinks also hide in those termite mounds, and so they have a ready supply of food hanging out in there. And so I've kind of thought about this, and I've actually started a couple colonies of geckos, some Binos geckos, as well as some of the morning geckos, and I haven't had the success that is probably needed to have a feeder gecko colony. So I haven't done a lot of this, and I've mostly resorted to trying to trick them into eating pink mice. So that's I guess when you're fighting nature, you're a lot of times gonna have a difficult time. Now, that said, I think all of the Antaresia genus are similar, where they hatch out small, they eat lizards for a long time, and then they convert over to mice. We did find a DOR children's python in Kakadu, and it had been, or actually it was south of Darwin a bit, Adelaide River area, and it had been hit by a car and it had a food bolus. So we, of course, wanted to see what it had eaten and pushed out this rodent, and it was an adult children's python, had a rodent in its belly. So, obviously they can have a lot more variety in their prey, but typically they probably eat rodents in the wild. I think the study by Shine showed that as they aged, they had more mammals in their bellies compared to reptiles. And that also gives the adults are not competing with the juveniles. They're not eating the same meals, and so they're not competing with each other for resources, and so that probably helps them make it to adulthood better, and a lot of different benefits of having that ontogenic change in diet. So I guess the easy answer would be, if you want to get your Antaresia feeding easily and reliably, feed them lizards. But then you run into the challenge of, who's going to buy a lizard specialist, from you if they need to feed on geckos or anoles or whatever. I don't know if they'll take anoles. So that's kind of the other question, if that is genetically kind of programmed in to recognize certain gecko species as food and not others, or certain lizard species and not others. But I guess that's difficult to say, without trying a lot of different things. It could also be individual preferences. Like some take to pinkies very readily and very easily, and others are just a nightmare. They will not take a rodent to save their lives, literally, So they are a difficult species, and I don't think there's like an easy answer to that. I don't think it's going to be easy regardless. Casey did say that once they reached a certain size, and I can't remember exactly what that size was, but they switched over to rodents much more easily. And I think too, a lot of times, like if you can kind of limp them through to the winter and they have a cool period and then it heats up again, they're a lot more apt to take pinkies after kind of a cool down period. But I've also found, like, they're not going to last that first summer, after they hatch through to the winter without eating. The problem with such a small baby is they decline rapidly. And so that's the problem that I've mainly had. And I can't say that I'm much more successful than you in that regard. Like, I lose quite a few pygmies in the year because I'll go on a herp trip or something, in the early fall or something, and leave them for a week. And like, once you start assist feeding, if you're trying to get them onto rodents, you can use tails or mouse leg drumsticks or whatever, just make sure you're not leaving kind of jagged bones if you're severing off a mouse drumstick. But I've used that successfully. You can kind of assist feed them, but you have to stay on top of it. You have to assist feed them weekly. Um, and a lot of times here in Utah, it gets really hot in the summer. My rodent production goes down because they're not in necessarily a thermally controlled room. And so I have, had in the past a lot of struggles trying to keep my rodent supply steady. So, but I've frozen a lot more, so I can thaw those and get tails or drumsticks or whatever pretty easily from frozen thawed mice. But yeah, you just have to stay on top of them, either feeding weekly or twice weekly. I do think they also like it a little warmer compared to some of the other babies. They do better if you keep them a little warmer. They're also more apt to feed on their own. If they're given higher temperatures, they're hungry, they're seeking something. So maybe putting a wiggling pinky in there will be taken more readily by a hotter snake compared to a cooler snake. So giving them a hot spot of in the mid 90s or something might be reasonable for a pygmy python. Again, they're very small, so you don't want to be given the whole cage. They want to be able to escape from that 95 degrees or whatever. So that can be a challenge as well. Having the thermal gradient in a small enclosure for a baby pygmy python.
95, that's interesting. I've definitely never brought it that up. I bring them up to 88, but I've never done 95, so I'd be curious. That's has that had good success for you, or have you noticed really a big difference, or they're just more apt to eating? That's really the only difference.
Right. And I've kind of chickened out a little bit in that regard too. I haven't brought it up that high, but I mean, they come from a very extreme environment too. It gets very, very hot in the summertime during the day. And again, a nighttime drop would be reasonable with those as well. But just having a small area where kind of a basking spot where they can reach those higher temperatures is reasonable. And then at night, it drops down into the 70s or 80s or whatever, low 80s. So that variation, I think for a lot of reptiles, that's reasonable, where they have kind of a hot spot at certain times of the day.
I guess my fear is that that high heat, their metabolism would be so high that they could also burn through their storage and basically like deteriorate within a week or two at that high temperature if you don't feed them. Like if I put them at 95, I'd be force or assist feeding. Like they're easy to assist feed. You put it in their mouth, they swallow it on their own. You don't need to actually push it down, but you'd probably have to do like twice a week at that temperature. No.
Yeah, exactly. That's kind of the pay or the exchange. If you're giving that higher heat, you definitely have to stay on top of them even more so. So yeah, twice a week, or as much as you can get out there, I guess. They probably, and again, in the wild, I'm sure 90% of the babies aren't making it to adulthood. They're getting picked off by something else, but I do think they are designed to kind of, get as big as they can, as fast as they can. And for a pygmy python, as big as they can is about 2 feet. So it's not really that huge of a snake. So, I think in the wild, they're probably eating fairly, consistently and readily during, shortly after hatching. The egg hatching usually coincides with a boon in their prey species. So lizards are hatching all over, and baby lizards are pretty dumb, so they're maybe an easy meal for a dumb hatchling snake too. They're not skilled yet. They haven't lived through the wild for very long yet. So, they need that kind of, I guess, easy meal from the environment. And I think they probably get more than they can eat certain times of the year, and then it gets hard, but that usually coincides with lower temperatures as well. So they can kind of find a spot where they can lower the metabolism and survive those times of low food availability, low prey availability. And that could also be during the very hottest part of the year too. So they definitely have the skills to survive those periods in the wild. And but I'm sure plenty get picked off by predators or just can't find enough to eat and die of the heat and things like that. So nature's pretty brutal. So, I don't know when you're dealing with a species that's largely a prey species, and just in general, like I think most species probably have a little bit of a loss rate in captivity, even when they have all the the stable environment and the unlimited prey availability to some extent. But so I don't know. Don't beat yourself up too much. They are a difficult thing to get started, especially if we're trying to force them to eat something they don't recognize as prey, at least as juveniles. Some of the tricks I've found that have been helpful are, using frozen thawed. Sometimes elicits a response. Washing the pink with like a unscented soap. And then just making sure to rinse it off really well, so you kind of get rid of that rodent smell. You can maybe stick a piece of gecko skin on their nose, or I've even I tried this with colubrids, where I'd put a piece of lizard tail in their mouth, and so the snake would smell that and start eating from the nose. And that was pretty successful as well. I again, I haven't had I've had limited success with a lot of these, but they've worked to some extent or another. I can't say there's one silver bullet that works really well for pygmy pythons. I have found that boiling, like frozen thawed pinkies, like thawing them in boiling water, works really well for wheat belt Stimson's pythons. Like they take off really quick with those boiled pinks. So that's one, I guess, silver bullet for wheat belt Stimsons. The pygmies didn't really go for them. They didn't really care about the boiled pinks. So I was kind of disappointed it wasn't useful for those. But
Have you
yeah,
thought about using chicken gizzards or anything like that in terms of assist feeding them at least?
I have actually, and I thought actually I was gonna look into getting some frog legs, because like I haven't tried this yet, but I know a lot of Antaresia in the wild hatch when kind of the frogs are coming out and having tadpoles and metamorphing frogs and things. And I know Stimson's pythons in Central Australia eat a lot of frogs. I don't know that pygmy pythons have really been studied too much, other than dissecting museum specimens. But I think obviously there's again that boom and bust in different species at different times, and so frogs obviously correspond with increased rain. And I don't know if that would work for pygmies, but I was thinking about trying it in Antaresia in general, because I think frogs are kind of an underappreciated prey species for Antaresia.
Yeah,
That
at
could
the
work.
very at the very least, you could at least try boiling and scenting or something like
Right.
that, right? Like
Yeah,
you could always use the frogs for other things as well. So
exactly.
multiple
Who knows?
uses.
Maybe that's the the silver bullet for pygmies. So I was thinking about taking like maybe just a small strip of frog leg meat and kind of into a mouse tail sized portion, and seeing if they would go for that. But
So I've tried the anoles. I've had 1 year I bought like 20 anoles and tried feeding them parts, tried feeding the animals, tried feeding them just how it doesn't work. I tried blending them, doing a full blender to be able to get the juices, and using the dip method of the pinky that's warmed up inside the juices after being washed. That didn't work. I did the whole same thing with the frog leg, going to your Asian market and getting all the weird different types of frog legs, and I've tried multiple. All that didn't work.
hmm,
The gizzards I haven't used yet, but after listening to all the Green Tree Python podcasts, everybody's always talking about the gizzards. So I wondered if it could be successful.
hmm.
Um, have you had any luck scenting with either chicks or either with quail? That's something that I wonder, because I feel like it wouldn't be there in their natural diet, but it seems to always work with everything else.
Right. I mean, yeah, some things like maybe don't make as much sense as, some people have tried like fish and put little fish in their Antaresia water bowls, and they'll go for fish sometimes. So I mean, I think, anything's possible. And so, I haven't tried it, and you know probably a little bit of like laziness, I'll just resort to assist feeding. And it's easy enough, like you said, for the most part. Once they kind of get the hang of it, okay, they're not trying to eat me. They're just putting food in my mouth. They usually finish what you put in there. Um, Now, I guess the downside to assist feeding is it's often like a piece of the mouse instead of the whole thing. I always thought drumsticks might be better than a tail because there's a little bit of meat, a little bit of bone, whereas the tail is just kind of skin and bone for the most part with very little meat. But there is some, and so I I don't. I started a study back in the day where I was assist feeding half of the pygmies with drumsticks and half with tails. But then you've got to factor in like the weight and all that kind of stuff. And it was a lot of work. And I eventually kind of gave up and just moved to whatever was easiest or whatever I had readily available rather than gathering data. So I kind of got lazy there. But that would be an interesting project to try again, or try some kind of feeding where scenting or something where you're documenting rather than just trying it once and abandoning it. You're giving it a more thorough shot and trying for extended periods of time, or assist feeding with different things. Obviously, if you're feeding whole prey, they're getting different gut contents and things from their prey species, or different bone densities. If you're just feeding pinky mice, it's kind of like a bag of fat, like they're not getting a ton of roughage, I guess. No hair, no bones for the most part. They kind of have weak little bones. So I don't know. It's a challenge regardless. Braining seemed to get them interested a little more. Like you can actually kind of hold the pygmy python in your hand and kind of tease it, or get it to smell the brained frozen thawed pinky, and that usually was something to get them interested with Antaresia in general. But then you're always having to brain the pinky, before you do it, and that can be a little nasty, I guess, but not too terrible. Basically, we're talking, getting a frozen thawed pinky, making a small incision in the head, and then kind of squeezing out a little of the brain contents there. And in a frozen thawed pinky, it's usually kind of soupy more than a freshly euthanized pinky. So, but yeah, that's had some success. I know, like putting it in a brown paper bag, overnight with a pink mouse might be another reason to just kind of give it nothing to think about but that pinky in front of it, and they don't really have a lot of things. Or maybe it's more of a naturalistic behavior that you're taking advantage of, and in the wild, they're not out in the open eating a pink mouse. They might be going into a nest or a burrow or a crack or something and trying to catch whatever prey is in there. So maybe that's simulating that environment and making them more comfortable to eat. So I know, like, say, a rubber boa is going to be more apt to feed on mice that are in like a nest type situation underneath something, rather than just handing them a frozen thawed, fuzzy mouse or something. They're going to go for the nest simulation versus just a frozen thawed rodent in their face, So I think if you can take advantage of their natural history, that might help a little more as well.
Oh yeah, I've definitely found a couple rubber boas with some actually probably that year's babies. I want to say,
Mm-hmm.
like, they're just out on the road at 1 o'clock in the morning. It's still 25 degrees Celsius out and in the mountains, and it's full of like
Yeah.
something, whatever it ate. It looked like it ate like 3 or 4 pinkies,
Oh, wow.
and it's just cruising across the road.
That's
So yeah,
cool.
no, they definitely like to eat multiples of whatever they're eating. Yeah.
Right. Yeah, they're cool snakes. Yeah, they're around here pretty readily as well. We see a few each year. Yeah, they're cool snakes. But yeah, it's I guess there really isn't like a trade secret or some kind of silver bullet for pygmy pythons. aside from maybe feeding them what they eat in nature, and breeding a bunch of geckos for the purpose of feeding baby pygmy pythons, and until they get to a certain size that they're willing to switch over to rodents.
It sounds like we're basically doing the same thing you're doing.
Right.
It's just a
Yeah.
challenge. My only question before we move on to a different subject is,
Mm.
at what point do you start force feeding or assist feeding? Do you wait a week? Do you wait first shed? Do you wait 3 weeks? I'm just curious, at what point do you decide?
Yeah, I usually wait. So I'll keep, once they hatch out, I'll put them in a container together, just drawer in a rack system, until they shed out, and then I'll split them up individually in their own enclosure, and then maybe a week or two after that. So probably a good month or so after they hatch, I'll start assist feeding. I don't think it hurts to start earlier. Probably right after the first shed, if you're just going to go into assist feeding, yeah, it's probably fine. I did my first assist feeding trials this last week with the first clutch, and I can't remember what date they hatched. I could probably look that up, but I did that last week, and half of them were kind of a pain in the neck. Like, they didn't want to eat it. You had to push it down almost to their stomach before they'd accept it and move on, And sometimes you'll get just some that just refuse, like just try to spit it out no matter what you do, and that can be a little frustrating. Got to be patient, play some calming music or something, while you're out there, if you're like me and very impatient. But I and really, if you have like a mouse tail, it's not difficult to put that into the mouth and then kind of work it down. You can almost just kind of slowly push it down. I usually wet it with something. You can just like a little bowl of water, and I'll kind of dip the tail in that so that it has a little bit of moisture to it while you're sliding it down. You don't want to try to slide some dry tail down their throat, because that could be damaging. I've had an incident where the bone from like a mouse drumstick broke through the skin. I don't think it was a pygmy python, but it was one of the Antaresia that it was refusing, and I was getting frustrated and kind of trying to force it a little bit, and it broke through the skin on the side of the neck, and that was not great. It healed and recovered, but it was, you don't want to have that happen either. So you gotta have some finesse, have some, patience, and usually, once they understand what's happening, they'll just start eating, and you can have them sit in your hand and finish off the meal. They're pretty good about that. So I don't think the assist feeding these guys is as challenging as maybe some other species, but it is definitely work, and you got to stay on top of it. I think that's the main secret, is just don't skip a week. If you start assist feeding, just know you have to go in there every week and get the job done, kind of thing. So if you leave town for more than a week, then you might be in trouble. You're probably going to lose a couple pygmy pythons, unfortunately. But so I always wondered why a lot of these professional breeders didn't go out herping more. But I think, if your livelihood depends on you breeding snakes and successfully rearing them, you can't skip a week. And some of these are a little more sensitive than others. So I can understand that now.
Cool, Michael. Do you have any other questions before we jump into the research paper? Do you have anything else you wanted to chat about?
I have a couple, but I have one about the gammon ranges. So obviously Paul Harris does have captive gammon range carpets. Mm-hmm. Has anybody been able to bring them into the U.S. yet? Or was it like after the whole U.S. Fish and Wildlife not letting Aussie stuff in happened?
I don't know. I mean, I imported some Antaresia from Great Britain in May, so I think you are able to bring stuff in. They actually labeled them as pygmy pythons, as Antaresia perthensis, even though they weren't pygmy pythons. So, but they still, got in, no problem. So, I don't know. I've heard it depends on the port they come through, and so I know Paul was asking quite a bit of money for them, so that might be a challenge to come up with that much. I would love to work with some. That'd be really fantastic, and they're really cool species. But it's a lot of factors go into that, I guess. If you want to risk such a high priced snake potentially getting turned away at the border, I think, of course, Paul would probably be better to work with than some like shady dude from Czech or something, like that smuggled them in or something. I think Paul would probably take care of you if they got turned back over to him. But it is kind of a scary thing to put up that much money to import something that may or may not make it. The other side of things, like right now in the U.S., a lot of federal jobs were, people were just fired. So I don't know that they have the manpower to really comb through every shipment. So maybe that's part of it too, is there's just not enough personnel to worry about, oh, is this snake legal or not? Or, is it something we should
Yeah.
reject? They don't have a lot of time on their hands. So they're probably just like, okay, fine. It looks fine. So they just go with it. The paperwork's in order kind of thing. So I wonder if that could factor into that as well. I don't know. It's hard to say.
Yeah, I've been waiting 5 months now for approval on Fish and Wildlife paperwork just to bring
Wow.
stuff from the U.S. up here for the Bahama boas.
Right.
And I think 2 months ago they came back and were like, "Oh, we just want a little more information on this from who produced the parents of the animals you're getting."
Right.
And the guy sent that off, and then we still haven't heard anything. So
Yeah.
I don't know.
No,
Obviously, it's probably just taking a lot longer now.
Right.
So yeah, nothing we can do about it.
Yeah. I mean, I brought over Western Stimsons from Europe, from Great Britain, and I produced them and wanted to send some back to Europe, and they denied the permits. And they say, we have no record that they ever have been legally exported. And I said, here's a copy of a permit that I got from you to bring them in legally. And they were even disputed, like they had some problem with them. And they got, they were actually came in as Morelia stimsoni. And they had to go back, and for some reason the British office was recognizing all Antaresia as Morelia, because at some point they were under Morelia for a brief period of time. So it's really a weird situation. So the Fish and Wildlife held on to him and said, these are not Morelia, what are you trying to do? And so I had to get the shipper to get some new permits, and with Antaresia stimsoni. But they came over as Western Stimson's pythons. And then, I had the paperwork, the stamp paper, and they basically said, well, just because we stamped them doesn't make them legal. I'm like, isn't that the only thing that makes them legal? Like, it just like makes you want to pull your hair out. And then they had issued Nick Mutton permits to send out Carinata over to Paul. And so I thought, oh, they're letting Carinata out of the United States if they're captive bred. And I had the same permits showing they were imported into the U.S. legally as Nick did. And so I applied for permits and sent them to the same desk. But apparently that guy had retired, and the person that had replaced him was a real stickler and I don't know if they were new or something, but they were just like, we've never issued permits for this species, and they've never been imported to the U.S. legally. And I said, here's a copy of, the permit and that they were brought into the country on, that I got them from, and here's, they're captive bred. Here's the documentation for that. Here's a copy of the permit that you issued last year for export of these from the United States. And she said, we have no record of that. And I said, I'm showing you the record. It's just so frustrating dealing with the Fish and Wildlife sometimes. So I just kind of gave up and said, okay, I guess I'm not exporting things anymore. I'll just send them, sell them within the US. But yeah, I don't know. Sometimes they just make it harder than it needs to be.
Yeah, I know for us, for Canadians, I went through a lot of the exporting and importing. And when I started exporting animals, I was told, just put everything under Morelia spilota mcdowellii. I don't care what it is, that is the one that gets approved. Everything else doesn't exist. It's all McDowellii. So, and I found it really weird, but then I went through and I talked to this, there's only maybe 5 or 6 people that do it in Canada, and it's all McDowellii. So for imports, we need the permits are from the other country right before they come in, and my understanding too is that's what's being done. And for the gammon range, if we want them in Canada, it's easy. You just need the money. Trust me.
Right.
Like, that's, if we wanted them, I could get them from Paul, and he would. Like, I've got my inlands from Paul, and that they came in. I could have easily had the other one. I just didn't want to fork out the $5,000 for a pair, which to me is just insane. I didn't want to.
Yeah, that's, I mean, It is a mixed bag for sure, and I think if I had the money, I'd probably do it. Just to have some. But yeah, they're next level. I just really like the patterns and colors that they have, and really cool species. But subspecies, whatever they are, I don't know. Really cool locality. We'll just call them that. But yeah, I maybe I fuss about that paper on the Antaresia, but maybe sinking Simpsoni with children I ruin fixes that problem. Now I don't have to export as Simpsoni, I export as children eye because taxonomically that's what they're scientifically shown to be. So yeah, I don't know. Sometimes we can take advantage of those silly things. Yeah, or I mean, even like any carpets. I mean, just sending them out as spelota. They don't really recognize subspecies for the most part. And since spelota come out of New Guinea as well, and they're imported into the pet trade anyway, like that's pretty easy solution to that because they're all spelota of some sort or another, except for imbricata and Brettles. So it seems like Brettles have gone over back and forth from the U.S. and Europe as well. So I don't know. It just depends on whose desk the paperwork falls on and whose port the snakes come in through. Because it's just a weird situation. I don't pretend to understand it. It's really frustrating though.
Yeah. Uh, I kind of wanted to get into talking about Agurnia, but I'm not sure if we want to dive into that whole thing with
Well, I'll
because we
say I'm
probably want
not
to keep it
much
down
of
to
much of an expert on Agurni. I've produced a couple
yeah,
species. I'm not that
yeah,
successful with them, and so probably save that for a better breeder of Agurnia than myself.
yeah. We just haven't. I don't think there's really been a Agurnia podcast yet, honestly. With
Right.
like, because I know I think Hussam keeps some, but he's
Mm-hmm.
only really talked about knobtails, and
Mm-hmm.
Alan has only really talked about his business stuff and hasn't really gone fully into Agurnia either. And he didn't even get to, like, I've rewatched those Brian Cusco
Yeah.
recordings
Uh,
of the rep of Herpeton,
yeah.
and obviously that was really cut short.
That
So
was
there's
that
really
was my least favorite
no,
part of that meeting was the fact that he didn't get the time that he needed to talk about a gurney. I was really upset by that because it was another speaker that went way long, and I'm like, come on, dude. And then Alan's like, sorry, I only have like 10 minutes to do this. I'm like, no, that's one of the big
yeah,
talks I wanted to hear. Yeah.
yeah, they're fascinating little
Oh yeah.
skinks. We have a lot of them available up here. Although I
Mm-hmm.
don't see the Strelata, but Mark at Northern Gecko produces like basically like Epsisolis, Hosmeri,
Mm-hmm.
Depressa, obviously.
Theos
What's
and Signitos and
yeah, I think
all
he
the
has
depressive
them on his
complex.
website.
Yeah.
Yeah, exactly.
Yeah, they're
Yeah,
very cool. Yeah.
yeah, they're super neat. All
They're fun to
right.
work with. I found a couple species in the wild, the Gidgies, the Depressa, a lot of the Striolata and some of their relatives, little Formosa and Pilbara. Yeah,
Oh
there's some
yeah,
cool Igurnia species for sure. I definitely love the spiny-tailed species. Yeah, those Depressa were really fun to find and just such cool little, different looking lizards for sure. Yeah,
yeah. They're pretty straightforward to keep. It seems like, although you
right,
just don't want to get them too cold, or is that
right.
not
I
really an
mean,
issue?
obviously well, in their natural habitat, it gets probably close to freezing in the cooler, but again, it warms up during the day, so they still have the opportunity to kind of uh, heat up to some extent, and then in the summer it's just blazing hot. I've been in there in the spring, and it's hot enough in the spring. I can't imagine summer. I went into like western eastern Western Australia in October, November, and oh my goodness, it was like 120 in the day, 115. It was just really hot. And that's Fahrenheit, of course. I can't remember
Were
what
they in a
Celsius is.
family colony when you saw them? Like, were they all doing their cool little family dynamic thing, or was it just a couple animals?
I honestly don't. I mean, the Gidgies, the Igurnia, Wait, what? Now it's failing me. What's the name?
Yeah, it
The
failed
Gidgeys.
me too.
Yeah.
uh,
The Gidgeys we saw like 2 or 3 in the same crack. So like they came out. It was kind of overcast that day that we were in their habitat, and I was really hoping to see them. And then like it looked like the sun was going to break through the clouds, so we said, okay, once the sun breaks through, let's give it 30-40 minutes to see if it heats up enough for them to come out. And sure enough, they started kind of peeking out of the cracks, and there were like a kind of a line, a big large crack overhang with some rock, and there were 2 or 3 of them kind of lined up in that crack, just kind of peeking their heads out and basking. So that was pretty cool to see. Stokes eye is the scientific. Yeah,
oh
Giddy
yeah,
Stokes eye. But
yeah,
yeah, so that was cool to see. And so otherwise, like the striolata were usually one-offs. The depressa, they're kind of in under bark and in cracks and in hollows in dead kind of trees, and so you'd see them in a crack. But how many are down that crack? It's hard to say. You see the one that's closest to the exit of the crack, but there might be more down in there. But we didn't see kind of like family groups in that regard. But yeah, I know in the rockier areas, it seems maybe it's a little easier to see. And I haven't looked for Eos. We did find Signitos, and we actually found a pair under the same rock in the same rock pile. So that was, I guess, another example of grouping or pairing in a small area. So yeah.
nice. That's pretty
Yeah.
cool. Yeah,
Yeah.
maybe the next time we get you on, we'll have you expand on the herping and the full Ligurnia care that you at least do. So eventually, we'll
Right.
hopefully get a couple people, to
Yeah.
cover that topic.
Lizards are a lot more work
Uh,
than snakes, that's for sure. So I love lizards,
yeah,
but I found out that I'm not as successful with the lizards as I am with the snakes, and I probably should stick to my lane and let better people take care of the lizards. But I don't know, they're really fun to keep. But I think in the future I might just kind of consider them more pets than breeding projects, because that seems to be easier on my mental state if I'm not worried about producing them or whatever. So they're kind of expensive pets at this point. I have produced a couple Hosmeri and a Depressa, but I haven't had any luck with my Striolata. But those are the 3 species of Agurnia that I keep. So I'm not, I don't have a huge collection of them. I have a pair of each, kind of thing. So that's about it. Again, somebody with more experience would be much better to talk to than me.
alrighty, Dominic. Do you want to get on to the final topic?
Sure. So obviously, you were a co-writer on this research paper. So do you want me to read the title, or just so people know, it's called "In Vitro Characterization and Antiviral Susceptibility of Ophidian Serpentovirus." So I read pretty much the entire 22 pages. I'll be honest, I probably understood about a page and a half of it. I found it really, really interesting. There's a lot of things that my mind was pretty much blown with. And but before I get into it, I'd love for you to just maybe talk about what the paper is, how you guys got into writing this, or kind of like the background story with it, and then maybe we can discuss a bit of the findings in there, because I find there's some things that, in my opinion, could literally change the way people treat their animals in that study, in that research paper. So I'll give you the mic there.
Okay. So this one was kind of an interesting project. It was a collaboration between myself and some folks at University of Florida, namely Stephen Tillis. I communicated with him quite a bit, and then Dr. Osibov. So they had isolated and purified several different serpentoviruses, or formerly known as nidoviruses, from different species. So we had an Antaresia serpentovirus, a green tree python version, and a ball python version. That those were the original species that the serpentoviruses were isolated from. So what the University of Florida group had done had found, they also had to develop a cell culture system to propagate more of these viruses. And so that can be a challenge, because viruses are oftentimes specifically tied to a certain cell types. There's different viruses that we, that are problem in, humans or whatever, that we just can't grow up in any cell culture, like they just don't have a way to grow them up, and there's not a really good animal model for them. It seems like they only infect humans, living humans, and not just the cells of humans or whatever. So it's a really kind of a challenge with some viruses. And serpentoviruses were somewhat challenging. They tried commercially available cell types that were, either birds, or they tried a lot of different mammalian cell lines and things to grow up the viruses, because you have to have something to work with, right? If you're going to try to test an antiviral compound, you have to have virus and growing virus to test that on. And so typically in this type of research, you would have a cell culture that supports growth and replication of the virus. And then once you've established that, then you can test different antiviral compounds to see if that slows down their growth or kills them, or stops their replication in some way or another. And so that's kind of the idea here. The folks at University of Florida in Gainesville developed the cell culture system. I believe they took a was it was actually from a diamond python. Yeah, they took tissues from a diamond python and isolated those cells, and so they had a cell system that would support the growth of serpentoviruses. They also tried a commercially available boa constrictor cell line of something or other. But the other things they tried didn't work, just the diamond python cells. I can't remember if it was heart cells or, anyway, a certain cell line from a diamond python worked. And so we, and honestly, we had a hard time keeping the cells from getting contaminated. Sometimes with these, when you're isolating cells from from tissue from an animal, they come along with other, bacteria or fungus or something like that. And so sometimes it's hard to get those all the way clean. But so, and also some are just more sensitive to introduction of bacteria from the cell culture technician, the person making more cells, because you also got to keep the cells growing. So you have the cells to infect with the virus, and then you isolate the virus from those cells to make more virus to use in the research. So that was kind of the first hurdle that they had to overcome. So the folks at University of Florida developed that cell culture system that would support growth of the viruses. They looked at the different growth kinetics of the different viruses and found maybe some were a little bit more readily, they more rapidly reproduce or things like that. So but they were fairly similar to each other. They showed that they grew better at different temperatures. So they looked at kind of the growth kinetics at different temperatures and how well they did. And so, they did all that kind of groundwork. And then once they had figured out, how to grow up the viruses, how to keep them happy and growing well, then they sent that assay to us. And then in our hands, we took and grew up and this is what we kind of specialize in, is taking viruses in cell culture, and then we add different concentrations of, drugs or antiviral compounds that so we can see if they inhibit the growth of the virus. So we took several of our positive control drugs that we use in our assays to evaluate, test antivirals in our system, in our antiviral testing group, to see if they had activity against these different virus isolates of the serpentoviruses. So we had tested, I think, I can't remember how many we started with, but we found that there were actually several that had good activity against the different viruses, and it seemed like all of the compounds had similar activity against the virus. We did see really nice activity, antiviral activity with ribavirin, which is, is kind of a broad spectrum antiviral. It works against a lot of different viruses from a lot of different viral families. So it often causes toxicity in humans and in other mammals. So it's hard to say, if that would be a safe treatment to use. This is very preliminary, just showing, the drug inhibits the virus in a very simple system of cells. Once you start, so that's kind of the first step you have to make to find a treatment, for these viruses. And so, I guess the first step was somewhat successful. We saw that ribavirin had really nice activity. A drug called remdesivir, you may have heard of it from the COVID outbreak, that was used against COVID and had some activity against the SARS-2 virus that causes COVID. Remdesivir worked, intermediately well. It looked like the green tree serpentovirus was a little more resistant to treatment, whereas the ball python serpentovirus was a little more easily treated by the drug, at lower concentrations. And so, and then the Antaresia python was kind of in the middle with that. That was at least with ribavirin, but with remdesivir, they basically had the same activity at the same concentration. And we actually didn't find kind of the lowest, I guess the lowest concentration of remdesivir we used was active against the virus. And so we didn't find the endpoint of activity for that compound. So that could even be as good or better than ribavirin in that regard. And then some intermediate activity with a compound that was discontinued because of some toxicity observed in some mammal species with NITD. It's called NITD-008. So that's from Novartis Institute of Tropical Diseases. They have a big antiviral development program, so they put forth a lot of them. We didn't see any activity with another broad spectrum antiviral called favipiravir. That one's typically not great in cell culture because it requires certain enzymes that are not present in a lot of cell lines. And so it could be, that's the other aspect of this, is like the drugs have to be metabolized to a usable form by the cells before they can inhibit the virus. And so that system in this cell culture might be lacking. We don't know. That's just kind of one of those black box areas. So favipiravir had no activity, but that could have been because the enzyme needed to convert the drug to its active form was not present. That's common with a lot of antivirals. There needs to be some kind of process of converting the drug to its active form. And viroxamine was also inactive, as well as pyrotavir and Infurgin, which was kind of surprising because Infurgin is just an interferon, but it's also species specific. So it's possible that it just doesn't work the same way in a reptilian cell line as it does in a mammalian cell line. So we identified 3 drugs that had reasonable activity against the viruses, and I guess a similar pattern where the green tree serpentovirus was a little more resilient to treatment than the others. That was somewhat consistent. So now that we kind of have those, some potential compounds for treatment, the next step would be to see if they're effective in an animal model of infection. Because like I said, it's one thing to, try to treat a virus growing in a cell. It's a very kind of simplistic replication cycle. Basically the drugs just kind of go through the cells and can interact with the virus. Whereas in a living system like a snake, you'd have to inject the compound somehow to get it into the animal, and then it has to get to the site where the virus is replicating. So it's a much more complex process. So this is kind of the first preliminary step in a long series of things that would be needed to show that these drugs actually could have a benefit in veterinarian clinic or something to treat an animal. So this was all crowdsourced for the funding through a carpet fest. They did a carpet fest in Florida and raised, $10,000 or something, $15,000 or something, and that supported the efforts at the University of Florida as well as our efforts to do this work. So it was kind of cool because no funding agency is going to fund this kind of research, and especially in the U.S. right now, we're barely able to get any funding for biomedical research, for human disease. So forget about snake viral diseases. Nobody's going to fund that right now, or very few places would. So that's kind of a nice thing. But at the same time, it's going to take a lot more money to fund animal trials and things like that. So I believe they have demonstrated that you can infect, clinic or experimentally infect a snake with different isolates of Serpentovirus and get that repeatable, disease phenotype that we'd expect from a Serpentovirus infection. That's been, I believe it was the same group with Stephen Tillis and Robert Osibov that did that, but my memory's not great right now. So, weekend of getting hit in the head by water polo players is kind of taking its toll on me, I think. But so, hopefully the work can continue in some form or another. I've heard rumors that there might be like a supportive or physiological way to treat the virus. There's some work being done with different temperature regimens, and they're looking at that as an option for treating Serpentovirus infection. And honestly, that would probably be the better route, and hopefully that's successful and they can find a method that works consistently to clear up a nidovirus or Serpentovirus infection. But right now, I think that's still in the experimental phase. They're trying to figure out if a reliable method that works.
So that is in the paper. They did do the experimental trials with temperature in the paper, and it does
culture. Yeah.
In the cell culture. So do you know that aspect? Are you able to talk about it a bit, just to say what the temperatures are? Or?
Not as well, but I mean, it's kind of tricky because, again, in cell culture, it's a very simple system. So, you could raise the temperature enough to kill the cells, and also kill. Obviously, that's going to reduce your virus. But how, again, it's kind of similar to the antiviral treatment. Is like, how much can you infer from a test in cell culture, to a living system, there's a lot that goes into that. But yeah, the viruses definitely grew at better or worse at different temperatures. And I guess I can't speak as confidently about those aspects of the paper as maybe Stephen Tillis could. And I know Stephen was on a recent episode.
Yeah, he was with
Stephen Cush, or was
it
not Cush?
I can check it out. I did listen to it, and
it was the expert in the idiot. That's
right? With Adam. Yeah, that's right. I was listening to that, and then they did ask him about the temperature treatment method, but I think it's still too preliminary, and I don't think Stephen was interested.
He went on
talking about that yet.
Yeah, he went on not another reptile podcast, and
that's right. Yeah.
He went deep into it, and basically he said, yes, it is, but I don't really want to talk about it. So the reason I wanted to bring it up is just, so he talks, if you look at the paper, it talks about bringing it to 95 degrees and talk about after a duration of 10 days, it killed off the entire cell culture. And I just found it interesting. Not to see if it is a future treatment. Obviously, it's just like you're saying the antivirals that potentially kill the virus. Now it needs to be tested to see if it worked, right? Mm-hmm. So I'd be curious to see if such a treatment, and we all know that whenever you're sick as snake, you put them in the incubator a little bit warmer, and if it's really sick, you put them in the incubator, you warm it up, and you give the antibiotics, and it's a miracle treatment, right? So now, could we do this for the virus? And looking at that study, it makes me wonder, and it makes me think that because serpentovirus is so widespread in the community, could there be a potential, maybe even cure or something like that for the future? And now, where I wanted to ask you was that a lot of people say, well, you got to be careful of what would be the aftermath, what could be the deleterious effects of that. And I'd be curious to see,
Right.
is there some with viruses? Because when it comes to bacteria, if you give antibiotics for just a few days and you kill off 95%, the 5% that isn't killed if you stop the antibiotics often turns into a super bug or a super bacteria, right? Is there similar effects that could be done by, let's say, if you put an animal in such temperatures but didn't kill off anything, what would be the negative effects of it? Do you know any of that, or am I going too deep?
No, I mean, obviously, like the strain to the system. If you're like you said, if you're increasing your temperature, you're also increasing your metabolism and other things. And if you have a weak snake that's sick and you're increasing its metabolism, that could have deleterious effects. That way, it could just push it to the point of almost no return. So yeah, it's a very complicated thing to so I think that's why they're hesitant, or they're still looking into it. They don't want to recommend something that's gonna end up killing a bunch of people's pets or something. That's not a great thing either. But I guess and I think we're still in the early phases of our knowledge in regards to serpentoviruses. It's hard to tell even which ones are more pathogenic or which ones are because we've seen a wide variety of from asymptomatic, like you don't even know your snake has it, but you get it tested and it's positive for serpentovirus, to the ones that are like just blowing bubbles and painting their walls with mucus, and they're just really sick, have a really extreme respiratory disease, to those that are sick for a few days and just die, and whole collections are just wiped out by certain serpentoviruses. So it runs the gamut with different outcomes. And so if you get tested and, oh yeah, I have a snake that's positive, does that mean you euthanize your collection, or euthanize that animal, or what do you do? It's hard to say. And some people have gone out and euthanized their collection because their snakes were positive, even though they weren't really symptomatic or having any ill effects of the virus. There's probably an effect of viral infection on reproduction. Sick animals typically don't reproduce, so that could have consequences for a breeding project. If you're have some sick snakes, they may not be as productive, and you might struggle that way. I think the nice thing, especially in regards to pythons, is it doesn't seem to pass from the dam through the egg to the offspring. And so if you can isolate the eggs from the mother and put them in a clean environment, you're probably going to have your snakes are going to be serpento free when they hatch. And so that's a nice aspect of keeping pythons. I don't know if that how that works with boids, and I don't know that. I mean, I think they've documented some boid species being susceptible to serpentoviruses, but I think it's typically a disease of pythons. But I think there have been a couple boas that have been shown to have serpentovirus as well. So, I mean, it's a very complex thing. We don't understand a lot about it right now, and I think obviously some people know a lot more than others about it. And Stephen Tillis would be a great one to talk to about for further information on serpentoviruses. We just need to get him to pause a little less. I kept thinking the podcast had stopped because he was taking these 20-second pauses between his responses. I'm like, are you there? Did it stop?
I was thinking the same
sorry,
thing when I
Steven.
listened to it.
Yeah. You got to get some snappier answers, Stephen. Come on, man.
Yeah, it's just
really great interview and really great information. So I think you just accept the pauses. That's all.
no, it was great. I enjoyed his. It's just like I've heard him talk about it. I've heard Nick Mutton say two sentences of the same thing. Like he said it on another podcast. You just you don't need to worry about virus. Just put him at 95 for 10 days. He said that exact line on one podcast. I'm like, man, it keeps coming out from different people. So
Right.
I'm just looking to see if there is more than if is this all derived from this paper, or has this information come from elsewhere? I'm just curious to see, because I mean, when I've read that paper, that's what it led me to believe. And again, I know it's just in a cell culture, but it gives hope for the future for herpetol culture, that there could be eventually some type of a treatment that maybe that could be used.
Right. I definitely think the paper in cell culture. I don't think you should rely on any information for treatment of a snake from a cell culture experiment. But I think there were a lot of whispers at Tinley Park in 2024, the fall, where I think this was discussed by a lot of people, because that's where I kind of heard whispers of it, was from I think it was Trap Talk. I was on the podcast with them over there, and they asked about that, and they said, "Hey, have you heard about this thermal treatment regimen or whatever? A week or two at 95 degrees kind of clears it up." And I'm like, I haven't heard that yet, and so I was kind of looking through the literature and trying to find out if that had been published yet, and then heard Stephen Tillis was involved, so I reached out to him, but I didn't hear back from him, so I haven't gotten, but then heard him on the podcast talking about it and saying that they're studying that now to see if there's a reliable and consistent way to do that. It's hard again to just say, hey, we had it work, so everybody go out and do this. It's just not very responsible to do that. I've kind of gone the rounds with Nick about his views on Serpentovirus, and I think he's kind of in some ways maybe putting his head in the sand, because it's just such a complicated issue. He wants it to be an easy, like, it's nothing to worry about. It's just some benign virus that doesn't really do any damage. But I think the data has shown, that there are some instances where it's extremely serious and whole collections die off. I have some friends that have lost whole collections of pythons to serpentovirus. So I don't think it's just like a non-issue. There's definitely something to it. It's very serious in some of the older ball python collections that got nidovirus or serpentovirus outbreaks and lost their whole collection. So it's definitely a real thing. Green tree pythons, it's a very serious infection in green tree pythons, it seems. They seem to be very sensitive to whatever serpentoviruses are kind of circulating in green tree pythons. So I don't think it's as simple as saying it's a non-issue. All you have to do is heat them up. Definitely, there could be definitely a lot more to it than that. So what the ultimate answer is kind of maybe between you and a competent vet. And that's another challenge, is a lot of vets don't know about serpentoviruses or don't even know they're a thing, and they might have to sit and do research just to try to figure out what it is or what's going on. Obviously people like Dr. Osibov or Dr. Tillis, they have a much better handle on that, and they would, but I don't think they're the kind of where you can pay them to give you advice on what to do with your snake. They're I know Dr. Osibov is a veterinary pathologist or something like that, where he has a lot of knowledge in regards to different diseases in reptiles. He's done a lot of cool research in that regard. So he would definitely be an expert, but I don't know that he has like he works at the vet College of Veterinary Medicine at the University of Florida. So obviously he's involved in veterinary research, but I don't know that he's taking patients or not, that kind of thing. And there's a few others like that are academics, but also are veterinarians. They might be able to give some insights into what you might do or help you figure out what to do with an infected collection or something. But it's definitely a challenge.
Yeah,
For
it's.
sure.
Yeah.
Sorry, go ahead, Mike.
From what it seems like maybe people took your guys' paper and kind of maybe inferred it without further papers, and been like, took your results and were like, well, 95 seems to work on this at this percentage and whatnot.
Right.
So maybe they just took your paper and decided to use that instead of going to a vet and stuff like that.
Well, I think that the research has continued, and they are looking at it
Yeah.
in an animal model, and I think that's what the kind of whispers were preliminary results from their
Oh,
animal
okay.
studies. So I think it's beyond the cell culture aspect of it, and I think that's what Stephen Tillis was kind of talking about on Not Another Reptile podcast that was talking about, serpentovirus infection and potential treatments for it. But he was just saying it's too preliminary to talk about right now. And that's the hard thing in science, is it takes time, it takes money, it takes, expertise, and we get impatient with that, or try to draw conclusions where, it's a little too early to make conclusions, or to suggest a treatment. So I think people are kind of just desperate for an answer. They just
Yeah,
want to
exactly.
do something to help their snakes, and I definitely sympathize with that. And I, like you said, that was always kind of the old school remedy anyway. Just increase their temperature, and that would probably help, and clear up a viral infection. I'm sure a sick snake in the wild probably goes out and basks more frequently, or tries to clear it up that way. But we also know that these viruses have been very deadly in the wild too. There was a shingleback serpentovirus that killed a lot of wild shinglebacks, and people were bringing these shinglebacks into veterinary clinics and things, and they had just severe respiratory infections and were dying in the wild as well. So you know it's not, again, it can be very devastating to certain species. So it's not as simple as saying it's a non-issue. There's definitely something to to worry about. You don't want to. And if you do have a sick snake, I mean, there's a lot of things you can do without a treatment, to prevent it from spreading to other animals. You always you put it in a different part of your facility, or you put it in a different room, or you work with it last or on a different day from the rest of your collection. As far as we know, they're not spreading through aerosol necessarily. It's mostly like through like saliva or whatever that's rubbed onto the wall or the glass of their tank or something, and then another snake picks it up by getting exposed to that. So if you're handling a serpentovirus, all of a sudden your hands or your gloves or whatever become the vehicle to transmit to another snake. So you're handling one, and then you're handling the next one and transmitting it that way. So looking at yourself as the the route of the vector of transmission could be an important kind of step in your head. Like, I don't want to handle a sick snake and then handle a healthy snake. You always handle the sick snake last, and then you're not going to necessarily transmit it that way. Obviously,
Definitely.
it is a respiratory infection, so it is possible to be spread through the respiratory route. And if a snake is housed together with another one, it can get sick that way. But if you've got snakes maybe in neighboring cages, it doesn't seem to spread, easily from cage to cage necessarily. So, and
Yeah.
again, that all depends on how they're housed, what kind of enclosure you have them in, and what access there is to respiratory, droplets or whatever. So
The paper
Your
went.
your paper also talked about the effectiveness of like bleach and chlorhexidine, I believe, on
mhm.
the
Yeah,
virus. So
That's
that
where I
kind
was
of
going to.
shows the ease. Uh, awesome. So that kind of shows people how they could at least minimize the spread, like you said, of the
right.
virus if they have it in their collection already, or they just want to possibly prevent future uh, spread of it in case they somehow got it or have it currently and they don't know if it's there. You can be
Mhm.
as sterile as you can, kind of thing. Try to
Right.
limit the spread.
Yep. Sterilizing your hands between, or even putting on fresh gloves, or cleaning your hands thoroughly between snakes or clutches or whatever. Obviously, it's much easier to kill a virus once it's outside of the body than inside of the body. So viruses are obligate. They have to use their host to replicate and to cause disease. A lot of disease of viruses is actually caused by an overreaction of your immune system. I always find it funny when people say, "Oh, we got to strengthen our immune system." A lot of the times, the disease is because your immune system is overreacting and too strong, and in that regard, in that language or whatever. So that's so it's not as simple as, like, because the virus it kind of hijacks your cells and uses the proteins of your cell to make more of them, copy their genetic material, make more proteins through that process, and then they bud through the cell and either destroy the cell or the cell just kind of lets them out. And so, they try to do that as quickly as they can. And they also have a lot of mechanisms to reduce the host's responsiveness to stop the viral infection. So there's a lot of different ways that viruses can overcome your immune system to trick it into thinking that there's no use in fighting it, or that it's part of your cell, so they don't kill the cell until it's too late and there's lots of virus released, and they move on to the next cell or whatever, or it's gone from the animal and into another animal. And so the virus wants to, what is a virus? And I don't know if it has wants or needs, but what they do is they replicate as quickly as they can and try to spread without viruses don't do well if they kill their host too readily. And so you don't see a lot of the more severe, like a Ebola virus is very easy to not catch because, typically you get Ebola through handling a person who has died of Ebola. And that's why it was such a problem in Africa, is their cultural traditions in some of those areas was that the family took care of the deceased. So they cleaned the body, prepared it for burial, that kind of thing. And so they were exposed to blood and body fluids and things. And that's how they were getting exposed to Ebola. And, it was a tragic, horrible thing. But if you just stop handling a dead body, you're not going to get Ebola very easily. Right. It's very difficult to get, and people get so sick that, okay, I need to stay away from that person. They're very ill. And so unless you're a healthcare worker that's working without protective equipment, you're probably not going to get Ebola very easily. So, the different aspects of, but typically, like we saw this with the COVID outbreak, is that as time went on, it got less pathogenic. People weren't dying, but it was spread very readily and very easily to people. And so that's typically how viruses evolve, is they get less pathogenic. That means they cause less death than they do, and so as they adapt to the new host. And so they are causing some illness, but a lot of times you don't see a lot of illness, but somebody can be carrying the virus and spreading it without even knowing it. And those are very effective viruses, right? When you're sick and you're showing symptoms, you got a runny nose or you have fever, that's your body trying to get rid of the virus from its own cells. And that's, so it's upping the temperature of your body, through a fever, and trying to flush the virus out, or get rid of it in different ways. A lot of the inflammation causes that heat, or that increase in temperature and things like that. So it's a very complicated system, and viruses are very good at what they do. They're very insidious and kind of get in and out without trying to be detected by your immune system, or but yeah, so it's a difficult problem to fight viruses, and especially veterinary viruses, where there's just not a lot of funding for research and trying to figure these things out. And we're all desperate for a treatment or some kind of thing to have happen. So it's really nice that people like Stephen Tillis and Dr. Osipov are actively engaged in this research and trying to find solutions for us. And I really hope they can come up with a good treatment for serpentoviruses, and I'm happy to help where I can if funding is available. But again, that's very difficult and expensive. Maybe we could supply them with some of our protocols and compounds that they could try it in their in vivo animal system, see if they could get an effective treatment in an animal model. But that's definitely the next step that kind of needs to happen before we can start saying what drugs are effective in treating serpentoviruses in snakes.
Yeah, it's very cool. So obviously it's kind of the fear of anybody that keeps pythons, right? And my collection, I try to bring very little because I'm so afraid of bringing anything that would infect everything. And from what I can tell, I've been keeping for a very long time. I feel like Serpentovirus has been in almost, I'd say 90% of everybody's collection forever for the past 30, 40 years. And I honestly think I've seen episodes of people losing animals or things like that. It feels like it's always been there, and most of them are just dormant or in very little concentration that it doesn't even affect the animals, right? Um, the interesting thing I found about reading that paper is that when it went into all the different types of things it tried to kill it or to eradicate it, it seemed like almost everything worked, whether it be alcohol 70 and 90, whether it be chlorhexidine, whether it seemed like every single thing they tried was extremely effective, right? Like, I don't even think there was one thing that didn't work.
Right.
I found that really interesting because it really feels like purely washing your hooks, washing your tongs, washing your hands in between a cage would literally kill everything and stop anything from passing from one place to another. I just found it interesting that it really showed that very basic cleaning solutions or very basic care would fix everything. Like I loved reading that because it really gave me hope that if ever anything got in, you could probably mitigate it pretty well.
Yeah, yeah, and I guess I kind of got stuck on a tangent there, but that was kind of the the idea behind my ramblings is that it's very difficult to kill them inside the animal, but once they're outside the animal, yeah, a lot of different things will kill viruses. Chlorhexidine or bleach or ethanol. Different things are very effective in killing viruses that are in the environment on surfaces or whatever. So one thing that kind of protects viruses to some extent in the environment is like the what, mucus or kind of the biofilm idea where you've got kind of a surrounding of mucus around the viral particles, and so the different anti or what's the term that we use for those? The virucides, uh, like bleach solution or ethanol, don't really interact with the virus because it's protected by that mucus. And then if a snake comes along and kind of slides its head through that mucus, that could potentially pass that on without being inactivated by some of these virucides. I'm giving the term virucides to the bleach solution or ethanol solution or whatever that we're using to kill the virus in the environment. The other thing to consider with these kind of treatments is contact time. Like a lot of things have a certain amount of contact time, so you don't just spray and then wipe it down and call it good. You've got to spray, let it sit, and wait till for example, ethanol, alcohol. Those things need to evaporate to have their activity on the virus in the environment. So if you don't let them have that contact time to evaporate and that they're basically ruining the protective layer around the virus, which is the membrane. So they have kind of like a lipid bilayer that surrounds the virus, the core proteins, and so that lipid bilayer is kind of like the surrounding of a cell, and the ethanol will actually the evaporation of the ethanol kind of ruins that layer, and that makes it so the virus can infect a cell. And so it basically kills the virus's potential to infect another cell and inactivates it in the environment. And so if it doesn't have that contact time or that evaporation, it has less activity in that virucidal mechanism. So basically you just have to leave it on there long enough for the material, and each type of virucide has a different contact time, and so that's all kind of dependent on its mechanism of action and how long it takes to accomplish that and permeate whatever the virus might be in.
Yeah, and I think it was interesting because it showed that ethanol 70% had a shorter contact time than the 90% or the higher concentration, which I don't understand why, but the lower percentage was more effective faster.
Right.
Which
right.
I thought was,
and that's what we commonly use in our viral labs. Like we have just bottles of 70% ethanol everywhere, and after you're done with something or you're finished with the treatment or something, you're spraying your hands with 70% ethanol pretty much constantly between everything you're doing, or, and then when you leave the lab, you're spraying 70% ethanol on your shoes, on your hands, on your, clothes or whatever, just to make sure nothing's traveling out with you. You're taking off all that protective clothing and autoclaving it. That obviously kills things, but you can't autoclave yourself. So you spray yourself with 70% ethanol. Yeah.
Michael, do you have anything else to bring up or chat about?
You're on mute. Yeah.
I don't really I don't think so. Not we need to, I guess, keep in mind Justin's been playing water polo all weekend, so we should probably let him get to bed sooner than later. But yeah, I was gonna hit on the contact time thing too, but obviously Justin hit that pretty well. So yeah, it's proper cleaning practices are definitely good.
Right. And I think there's kind of general rules. So if you just kind of look into, whatever you select for your viricide, say you want to use a 70% ethanol, just look, just do a Google search or something for contact time for efficacy of 70% ethanol or something like that. Contact time for 10% bleach solution to have its maximal effect on, different pathogens or whatever. You could use that for a lot of different things. It'll kill a lot of different things. So that's probably the easiest way to do this. I don't have those numbers off the top of my head. We don't do a lot of work with viricides. We're mostly trying to find treatments that will work in a person or in an animal to clear them of virus.
Awesome. Alrighty, so I think we should.
Oh, go ahead.
Do you have anything,
I was
Dominic?
going to
Or
say
should we,
before
try to
we
close
end
it
it, I
out
was
here?
going to ask you one thing. Did you how's it going with your blackheads? Did those eggs hatch? I think I heard that you only have one that came out. Just tell us a bit about that,
Yep.
and then we'll.
Unfortunately, I still am missing something because the eggs were good. They went full term. Like I had 5 die in the egg, which just frustrates me to no end. They didn't hatch out for some reason. When the first one pipped, I even kind of opened some of the other eggs and they just weren't moving. So I don't know what's happening, what I'm missing here. They definitely weren't too wet because they were pretty sunken in towards the end. And the one that hatched was in, one of the eggs that was fairly sunken in. So, I don't know why that one hatched and the other ones didn't. It's just kind of driving me nuts here because I felt like I did the stuff on the front end to be successful. And the fact that they all went kind of full term gives me some hope that I'm on the right track. I talked to a few people, there was a suggestion that maybe it's an elevation thing. Maybe my elevation's high, but I don't know, it's some of those things where like, you don't know if it's just that herp mythology that kind of accompanies things or what's going on. But yeah, I'm very frustrated why I only got one egg to hatch, but the baby seems to be doing great. Now they're very similar to pygmy pythons that they're very difficult to get feeding. So I'm starting, assist feeding with with that one as well. And I still have 2 from not last year, but the year before that are doing well. They were the ones where I had the incubator malfunction. So they hatched out with their small eyes. I think that's just a environmental thing. I don't know that it's necessarily is going to be genetic or anything like that, but
spiking up temperature or down temperature, do you know? Up.
Yeah, the room got up to like 100 plus and just, yeah. And so the eggs were exposed to extremely high temperatures early on in incubation. So, and I had 2 hatch out at that time. So, I thought I did everything right this last time and I only got 1 to hatch out. So I'm not sure, what I'm missing still, but hopefully I can figure it out. I'm going to try maybe a couple different incubation techniques, move them into 2 different nest boxes and see if I can figure it out. But yeah, I'm a little frustrated at this point, but definitely not an expert with black-headed python incubation.
I'm just curious. I've never gotten there. Just by curiosity, when you see the first head pip, are you slicing just a little tiny, like all the eggs, just a little bit? And did those five babies actually move in there, or did they already look stillborn? Or
Yeah, I'm a little gun shy with cutting eggs. I went in and cut some Woma eggs a little early just to, I don't know. I think I was breeding ball pythons at the time, and that's just what you did. You open the ball python to see what kind of morphs you got or whatever, So I was doing the same thing. And like all the Womas that I opened the eggs early, they all died. They didn't make it out. And the ones that I didn't came out just fine. So I think, I'm definitely gun shy about opening up Aspidites eggs early.
are you doing a large barn door or just a little tiny
No,
little
no.
line?
Just like a kind of a V, just kind of pinching up the eggshell and cutting just a small opening so they can escape. I'm not cutting off the whole top of the egg like some of those retic breeders, but so that's uh, so I don't know. I'm just kind of an idiot trying to make my way through this, but so something went wrong and I failed again and just got one this season. But its eyes are regular size and it's feisty, happy little baby. So hopefully I'll get it feeding, and it's a female too, so that's not a bad problem to have. Sometimes I wonder too, like I got the adults, I got the original pair as adults, so they were used to a different environment, and sometimes I think raising up babies in your own setup and situation, and whatever yearly cycles go through your own collection is oftentimes better than or acquiring animals as juveniles is probably the better way to go if you're starting a breeding project. Not to say that you can't have success with adults. I mean, obviously I got these adults and I'm hatching out some, but I think your chances of success go up if you start with juvenile animals. So I think I'm going to hold some of these back and see if I can get some replacement, maybe swap out, have some bloodline swaps with other Western blackhead breeders. But I had an original trade lined up with Lucas back in the day, but the animal that I had ready for him ended up not surviving, not thriving. So lost that animal. So that was a little bit of a bummer. But yeah, one of these days I'll have to get a swap with Lucas going. He's got some, or he did produce a couple nice ones, and then I think he's had trouble getting eggs after that. So we'll see. But he's knocking it out of the park with everything.
I was going to say he's got a lot of success.
Yeah.
A lot of success. Good for him. Yeah.
He has 2 diamond clutches this year.
Yeah.
And he got green trees
not too long ago. But yeah, green trees, and yeah, he's killing it. Yeah.
He's beating the Podfathers to the punch with that.
Yeah. Such a nice kid too. So I mean, I'm really happy for him and his success. He's just a great guy. Yeah.
Well, sorry to hear about your blackhead, but keep at it. You're going to get there. You're a step further.
I'll figure it out one of these days.
You'll get it.
Yeah.
Perfect. You want to close it up there, Mike?
Alrighty. So, Justin, do you want to give everybody your details and how do you prefer to be contacted and how they can reach you?
Yeah, I mean, I have a website. It's not updated very often, but it kind of shows what, for the most part, what I'm working with. There's a few projects that have kind of fallen off, and I'm not really actively breeding coastals or green trees or things like that anymore, but they're still on my site. Mostly Antaresia and a few different carpets right now, and maybe an occasional lizard here and there. So Australian Addiction Reptiles, my website's australianaddiction.com. You can find me on social media under JG Julander or Justin Julander. JG Julander on Instagram, Justin Julander on Facebook. And, if you want to email me or text me, I can get back to you on what I have available or that kind of thing. So yeah, thanks for having me on. Oh, also, I guess I can throw a plug out for my own podcast, Reptile Fight Club. We've been kind of slower during the herp season, we'll go out on herp trips. Usually I'm with Rob on herp trips, and occasionally we've recorded from the field, which is kind of fun to do, but haven't done that lately. But yeah, if you want to check out that, we kind of take two sides of a topic and flip a coin, and whatever side you randomly get from the coin toss, you got to kind of defend that, even if you don't necessarily agree with it. So yeah, it's been fun. We just had Ron St. Pierre on, so that should be coming out next week, and yeah, good times.
Awesome. Glad to. I know we both enjoy that podcast when it comes out, so we always enjoy Rob's input on things because he has some great questions.
He's brilliant. And I really really enjoy recording with Rob. It was fun with Chuck. I miss Chuck, but
Chuck was fun too.
Yeah.
Hopefully eventually he makes his way back, but we definitely love Rob too.
Yeah, Rob stepping in has been fantastic, and he's great. Just really knowledgeable and really fun to record with. I enjoy herping with him as well. He's great. He was just out here for a visit, so we got to find some rubber boas with Rob, so that was fun.
Awesome. Gotta click those off his life list.
Right. Yep.
Alrighty, Dominic, you want to say your stuff?
Yeah, I was say definitely a great podcast. I've always enjoyed listening to it. Justin, I just wanted to thank you for coming on. Really, really appreciate it. Appreciate you donating the time and coming on. Wealth of knowledge. Love it. Thank you. For myself, domsreptiles.com. Same thing. It's a website that shows my information. Horrible at updating it. But you can see my collection what it looked like 6 or 7 years ago. It has changed a lot, but it's got my emails and my information. Facebook is just my name, Dominic Cavano, and Instagram is Dom's Reptiles. And again, thank you, Justin. Really appreciate it. And Michael, your turn.
Alrighty. Well, thank you, Justin, for joining us. I definitely appreciate you coming on. It was definitely a good first episode with a guest for us. You were really good at explaining everything that we asked, and we didn't have to give you too many questions for you to give us some really good answers and really in-depth ones. So yeah,
you just have to tell me to shut up sometimes. I tend to talk. But thanks for having me on. It was an honor to be your first guest. Thanks, guys.
It's always good to let the guests say what they want to say because it's usually really good. You can find me on Facebook and Instagram at Michael Gillen, and you can also find us at Reptile Rejects on Instagram. Alrighty, guys. You guys have a good one, and we'll be out next week with Chris Painchab. Thank you, guys.